Good morning to all. We will start our today's session. Today, for the first session, we have with us Dr. Amrutlal Patel. He is a scientist D and Joint Director of Gujarat Biotechnology Research Center, Ahmedabad, India. So, here, previous to his he has done his BVSc from veterinary science in veterinary science in Gujarat Veterinary College, and his master's from Veterinary Research Institute, Itzanagar, UP, and his PhD he has done from in Anand Agriculture University. He also worked as a veterinary office in Baroda Dairy, and he also served as a scientist in Zydus Research Center, Cadila Healthcare, and research assistant and vaccine and infectious disease organization, Canada. He also served as a research associate in Department of Animal Biotechnology, Anand Agriculture University. He worked as a consultant for Zellaris Labs, Anand Agriculture University, and principal scientist R&D, Hester Biosciences. And he did his also research associate at Gujarat Biotechnology Research Center, and he has done his postdoctoral fellowship at the Vaccine and Infectious Disease Organization from International Vaccine Center, Canada. He has published. He has 2 patents to his credits and published more than 45 papers, more than 50 papers in international and national level journals. He has 4 book chapters to his credits, and he has 10 GenBank submissions to his credit. He has presented in more than 10 conferences, like abstracts and poster presentations. He has participated in more than 12 seminars and workshops to his credit. So with this short introduction, I invite Dr. Amrutlal Patel to deliver his lecture on applications of biotechnology to veterinary medicine. We welcome you, Doctor. Can start.
Thank you, Doctor, for the introduction. Good morning, everybody. Is my screen visible?
Yes, Doctor.
So good morning, everybody. Today I will be presenting an overview of applications of biotechnology in veterinary medicine. So I will just give a brief definition of biotechnology and veterinary medicine before going for the applications. As all of you know, that biotechnology is the area of biology that uses living processes or systems or part thereof to manufacture the product or technology intended to improve the quality of life. And this is a broad definition, and as you know that there are many domains or disciplines where biotechnology is applied, including animal science, animal biotechnology, medical biotechnology, plant biotechnology, industrial biotechnology, environmental biotechnology, and marine biotechnology. And if you are looking for a place where all this work in all this domain is going on, is Gujarat Biotechnology Research Center. At present, I am working in GBRC, and this is the place where all this biotechnology disciplines related work is going on. And to give a brief introduction about veterinary medicine, is a branch of medicine that deals with the prevention, control, diagnosis, and treatment of disease, disorder, and injury in animals. And many of you must be aware that there are diverse range of animals which are included in the veterinary medicine, including cow, buffalo, sheep, goat, horse, donkey, camel, elephant, dog, cat, lion, tiger, other wild animals, poultry, as well as other birds. So you can understand that there is a huge diversity in terms of the patients we get in the veterinary medicines. They all have different anatomical structures and different physiology. Even if you go for the medicine, the different animals have a recommendation of different medicines. Even doses also will be different for the different animals. Even animals with the same size also will have a different doses, and some medicine will work better in one animal but not in the other animal, like that. So many variations are there. So veterinary medicine is quite a challenging field for the those who are looking for the pursuing the veterinary medicine. But at the same time, it is not as much advanced as human medicine. Like some of the things which like heart surgery or like that, we are not doing in animals. So on the other hand, the scope or the depth is less compared to human medicine. So, when talking about the biotechnology applications in veterinary medicine, you can develop vaccines, diagnostics. There are technologies to enhance the animal productions. There are assisted reproductive technologies to improve the reproductive performance. Then there is a recently emerged field metagenomics that you can apply in number of areas, including human metagenomics, gene therapy, especially in the pet animals, then induced pluripotent stem cells. That is one of the technology which is explored in the veterinary medicine. Then recombinant proteins and antibodies, which are used for as a biopharmaceuticals. There are number of human products are there in the market, like biosimilars, and number of companies are working in this direction. And in veterinary medicine also there is a requirement of this recombinant proteins and antibodies for developing vaccines, diagnostics, as well as therapeutic proteins. Then there is one more area like transgenic animal bioreactor, where you can do a farming of the biopharmaceutical molecules in animals. Then there is a one more area is introducing the disease resistance in the animal. They like identifying some disease resistance gene in the animals and then introducing in the other animals who are susceptible to it. So these are the broad areas which I will cover today in my talk. Apart from these broad areas, there may be more applications in the applications of the biotechnology. So vaccines, as most of you are aware now, because of the COVID, that vaccines are the substance which is used to produce the antibodies against the target pathogen. And this substance can be the pathogen or its part or the synthetic. And it basically acts as the antigen without inducing the disease. So there are a number of diseases in the veterinary medicine or in the animals. I have listed just few. Those are very much important, like brucellosis, foot and mouth disease, tuberculosis, rabies, avian influenza, bird flu, infectious bursal disease, Newcastle disease. These are in the poultry, sheep pox and goat pox. The lumpy skin disease, anthrax, leptospirosis, canine distemper, babesiosis, trypanosomiasis, theileriosis. Many of these diseases are zoonotic diseases. They also occur in the humans for by transmission from animals to humans. So they are very much important. And in terms of vaccines, you know that traditionally vaccines are made, the conventional vaccines are made by inactivating the pathogen or organism, that is killed vaccine, and attenuating the organism by passaging in the non-natural host. Like chicken embryo, you passage the pathogen in a chicken embryo for multiple passage, like 40-50 passages, the organism loses its virulence and that doesn't cause disease anymore. So that is the category was the live attenuated vaccines. Those two type of vaccines were produced earlier days, before 2-3 decades. Nowadays, new generation vaccines have arrived like viral vector vaccines, DNA vaccines, protein subunit vaccines, multi-epitope vaccines, and mRNA-based vaccines. Most of you must be aware of these words because of the COVID, especially mRNA vaccine and DNA vaccine and viral vector vaccine, that is Covishield, which is made by Serum Institute of India. DNA vaccine is made by Zydus. mRNA vaccine has been made in the foreign countries. So the protein vaccine is made by the biologically. So there is still no multi-epitope vaccine for the COVID, but these are the different category of new generation vaccines which have been developed for various animal diseases, and they are in the market. So this is the flow chart of how we design the vaccines, how to identify the gene is by the sequencing of the genome of the pathogen, then identify genes which are inducing the antibody production or cell-mediated immune response, and then go for cloning of those genes, make the cassette to make either DNA vaccine, subunit vaccine, that is protein-based vaccine, or the viral or bacterial vector-based vaccines. So this is one flow chart of right from the sequencing to the clinical trial. For example, you have any pathogen, then you have to sequence the genome of the pathogen and do the bioinformatic analysis to identify the vaccine targets, which antigen, which gene protein can induce the antibody response or cell-mediated immune response, and then those gene you clone in the vaccine vector, either in the plasmid vector or viral vector. Then one of the way is to purify the proteins and use as subunit vaccine. Then do initial preclinical study in the mice model, where you study the safety as well as efficacy, like how much antibody response is generated or how much dose is tolerated by the animal. Those type of studies are conducted in the mice model, and then those sera we study for the antiviral activity against the pathogen, and then after studying the efficacy, we finally go for the clinical trials in the humans for studying the safety, efficacy, and potency. So these are the number of different ways and number of different vaccines produced in the veterinary field. The first category you can see is the plasmid containing gene for target antigen. So this is basically the plasmid DNA vaccines. There are some examples in the plasmid DNA vaccines. Then there is another category of vaccine is the protein-based vaccine, where you first clone the gene in the plasmid and transfect in the suitable cell line, either in the different type of host. You can produce protein in the E. coli or insect or mammalian cells, and then purify that protein, and that protein you use as a vaccine candidate. So there are a number of vaccines, protein-based vaccines in the veterinary field. Then in the same similar type of vaccine, you combine the multiple proteins. Instead of only one protein antigen, you can use multiple protein antigen to provide broad protection against the disease. So these are called as chimeric protein vaccines. Then there are chimeric viral vaccines, where your antigen is expressed on different virus protein. So the vector virus is having its own protein as well as the antigen of the pathogen. So these are the number of vaccines in the chimeric viral vector. And there is one more category where antigens are not present on the virus coat, but it is encoded by the genome of the virus. So like adenovirus vector-based vaccines, your foreign gene is integrated in the adenovirus genome. And from the genome of the adenovirus, when it is infecting the cells, your target antigen is synthesized. So these are the broad category of vaccine and different examples of these vaccines in the veterinary field. This is the list of the vaccines which are available in the market against the different animals, against different pathogens. These are produced, I have taken only one category, viral vector-based vaccines. You can see that number of pharma companies like Boehringer, Pfizer, Zoetis, Merck, Seva, these companies are producing these viral vector-based vaccines against for the species like canine, that is dog, feline, that is cat, equine, that is horse, swine, that is pig, bovine, that is cattle, buffalo, and avian, poultry. Even there are vaccines made in the wildlife species and rabbits. So against the different pathogens like canine distemper, feline leukemia, rabies, equine influenza, West Nile virus, circovirus, number of different pathogens, animal pathogens against which these vaccines are made, and they are commercially available in the market. So another area where we can think of in the veterinary applications is the diagnostics. So diagnostic earlier days, the conventional method were used was bacterial or viral isolation to identify the which pathogen is there causing the disease. But nowadays, because of the biotechnology based tools, we can directly identify the pathogen by screening the different antigens, that is proteins antigens of the virus or bacteria, or antibody against that antigen in the host, or the nucleic acid, DNA or RNA of the virus or bacteria we can identify by different tools and diagnose the disease. Apart from pathogens, we can also use these biotechnology based tools to identify the genetic diseases in the animals. So these are the different types of diagnostics available in the market based on the PCR or real-time PCR. As most of you know now that real-time PCR and PCR, RT-PCR and PCR were used for the diagnosis of COVID. So similarly, such tests are also available for diagnosing the animal diseases manufactured by different companies like ID Thermo Fisher, UBio, this Symbiotics, IDEX, number of companies are having this PCR and real-time PCR based diagnostic test as well as ELISA based test. ELISA can be used to detect the protein antigen or the antibody against that particular diseases or viruses or pathogens. Then recently there are new type of assay coming up is the rapid assays, lateral flow based assays that detects the antigen within a few minutes, within 5 to 10 minutes. You put a drop of sample in the strip and then add some buffer and let it flow through the cartridge, and you will see the bands against different control as well as the specific pathogen. For COVID also, number of people might have seen this type of lateral flow strips where it can rapidly detect the antigen of the COVID virus. And similar rapid antigen based kits are available for the number of veterinary pathogens and produced by the UBio from the one of the Indian manufacturers. Apart from this PCR, ELISA and lateral flow kits, even next generation sequencing can be used to diagnose the pathogens. And as you know that the first COVID virus was identified by NGS technology when there was outbreak in the China. The technique which was used to identify what was the cause of the disease was the next generation sequencing, because you don't have any knowledge of which type of virus it is. So in that type of cases, you have to go for the next generation sequencing, where you sequence all the nucleic acid present in the sample, and from that sequence you identify which pathogen it is. Also, there are microarray based diagnostic tests are available, especially screening for the number of copies of the gene or the chromosomes, especially aneuploidy diagnosis or deletion duplication type of diagnosis in the especially in the prenatal diagnosis type of samples. Then there are technologies to enhance the animal production which can be used for the animal production. See, traditionally, there was a selective breeding type of method was used in which the animals which are performing better than the average were used for the breeding. Male and female animals which were producing higher milk or meat production were used for the breeding. But it was taking very long time. Of course, so far whatever development has been made, that is through selective breeding, and milk performance has been improved several fold by selective breeding. But recently we have new technologies for improving the performance as well as introducing the new characters by transgenic animals production or knockout animals generation or by genomic selections. So this is one of the example. As you can see that double muscling type of phenotype in Belgian blue cattle due to the mutation in the myostatin gene. And this mutation in the myostatin gene is tolerated by the animal, and animals live the healthy life. But the muscle mass is double compared to the normal wild type myostatin gene. So this provides a hint that if we mutate myostatin gene or inactivate myostatin gene in other animals like goat or sheep, we can increase their muscle production. So with this objective, we have worked on myostatin at Veterinary College Anand for a few years, and we published number of papers on myostatin knockdown by short interfering RNAs in the in vitro cell lines. And there was an objective to generate the stable cell line. We generated stable cell line, and then there was a plan to produce the animal from using the nucleus from the stable cell line, but that has not been proceeded further. The project was over. The another area is the genomic selection. Traditionally, it was done by selective breeding, but through genomic selection, what we can do is we can scan the entire genome of the animal for millions of SNPs, and those SNPs will have some phenotypic associations. So initially, to identify which SNP has a association with the performance traits, we screen the reference population, thousands of animals we screen and record their performance like milk production, meat production, and we screen their SNPs, and we identify that which SNP has association with different traits of the production traits. And using those association, we derive the equation. And after deriving the equation, finding association, we can study any study population right from the birth of the animal. We can identify whether that animal is going to give the higher milk production or not. So this is one of the latest advancement in the biotechnology, where genomic selection is used to identify which animal should be used for the production purpose or for breeding purpose right from the birth. Then one more area is the assisted reproductive technologies. In that, number of technologies are there, like artificial insemination, multiple ovulation and embryo transfer, semen sexing, IVF, ICSI, intracytoplasmic sperm injection, embryo sexing, somatic cell nuclear transfer. These technologies are covered in the assisted reproductive technologies, and they have been used for enhancing the animal production. Artificial insemination, as you know, that the semen from the high potential bull is collected, and it is prepared in the doses, and these doses are used for insemination of the animals to enhance the performance of the progenies. And there is another approach which is called semen sexing, because in animals, as you know, that 50% are male and 50% are female offspring or progenies. And all male are not used for any production or production purpose. So many of the males are slaughtered. Especially cow are considered a sacred animal. So in India, if we prevent the slaughter of male animals, male cows, then it can be a better option. So there is a technique called as semen sexing, where we can separate the spermatozoa based on the X and Y chromosome, because X chromosome has a little bit more DNA compared to Y chromosome. We can stain them with DNA binding dye, and the difference in the DNA size gives difference in terms of charge. So based on the difference in terms of charge, we can separate the male sperm and female sperm in two different tubes. And these female sperms, now we can do the artificial insemination to produce the female offspring or female calf, so that we can prevent the slaughter of the male calf, and thereby we can increase the production. And this technique is currently used even in India also. Then there is a intracytoplasmic sperm injection and IVF. This is mostly used in the human subjects, but we also do it in animal to improve the or enhance the utility of the females which are producing a higher production. So in the classical IVF, you put millions of sperm in the oocyte together, and then there will be natural fertilization. The one sperm can enter into the oocyte. But in ICSI, intracytoplasmic sperm injection, with the help of microscope, micromanipulator, we pick up one sperm and inject into the embryo or oocyte of the animal. And after this, it will be developed into the embryo, and it will be transferred into the animals. Then there is one more technique, that is embryo sexing. So once embryo is developed, we can identify whether it is going to develop into a male animal or female animal. So based on the X and Y chromosome specific markers, we can identify whether the embryo developed is going to be male or female using the Y specific PCR. So we can here also we can use the sex embryo to transfer and prevent the slaughter of the male animals. This is the technique of multiple ovulation and embryo transfer. We can use the superior female to get the more progeny out of the animal. Normally, one animal will give 10, somewhere around 10 calf or offspring during its entire lifespan. If we consider 1.5 years for one generation, then we can get only from a good animal, we can get only 10 offspring. But if we do multiple ovulation, we from the good animal, we can generate hundreds of or thousands of embryo and transfer those embryo into the other animals which are not producing higher as compared to this animal. Then we can, from the same good animal, we can produce thousands of offspring in the entire lifespan. So we can enhance the production of the animals through multiple ovulation and embryo transfer. This is the flow chart of the cloning, that is somatic cell nuclear transfer, that is first used for production of the dolly, the cloning of the sheep, where what scientists have done is they have taken the mammary gland cell from the fin dorsitae, which is white facing, and from mammary gland cell they have taken the nucleus, and this nucleus they have transferred into the oocyte of the Scottish blackface eel. So from this oocyte, they have removed the nucleus, and they took nucleus from mammary gland cell, and that mammary gland fibroblast cell were fused with this enucleated oocytes. And from this, they have developed embryo and transferred into the surrogate mother. Surrogate mother was a third type of animal, which was not neither related to this Scottish blackface eel or the fin dorset eel. And from this, they got the offspring, which is identical to the fin dorset eel. So that is called as cloning. So this cloning can be used to enhance the production of the animals which are genetically superior. So one more area is the metagenomics. Metagenomics is basically a collection of the genetic material or a study of genetic material from the community. And especially in the animals, rumen has a rich source of microbes. If you look at the rumen is basically a part of the stomach. In animal, ruminant animal livestock, there are four compartments in the stomach: rumen, reticulum, ovomeson, and abomasum. So rumen is the first stomach, which is quite big, about 25-30 liters capacity. And there are number of bacteria, protozoa, fungi are present in the rumen, which do the fermentation of the grass, because animal eat grass and some grains also, and these are very high in fiber and very difficult to digest by the enzymes. So bacteria and other protozoa present in the rumen actually do the degradation of this grass, which are eaten by the animals. And this bacteria or protozoa have the very rich source of enzymes, which has a high potential in the industry. So we do the community analysis of this bacteria present in the rumen through metagenomics. So in metagenomics, we isolate the DNA from the sample or community, and this DNA we fragment it and sequence it. And after sequencing, we do assembly and annotation to identify the presence of different types of bacteria, as well as presence of different types of genes. So we after identifying the different genes responsible for degradation of this grasses of fodder, this enzyme we can clone and express in the laboratory, so that they can be exploited in the industry. So at Veterinary College Anand, we worked extensively on the rumen metagenomics and published 42 papers over the span of 10 years. And out of that, we published 4 papers for the enzyme isolation from the rumen microbes. And we also filed 1 patent for the enzyme isolations from the rumen microbes. So one more area is the gene therapy. Gene therapy is basically a tactic to introduce the functional copy of the gene or the modified copy of the gene, which with a aim to treat or cure the disease. And this can be done by replacing the disease-causing gene with a healthy copy, or inactivating a disease-causing gene that is not functioning properly, or introducing new or modified gene into the body that can help to treat the disease. So in gene therapy, there are 2 types: ex vivo gene therapy and in vivo gene therapy. In ex vivo gene therapy, you take out the cells from a patient, and in those cells, you do a genetic modifications in vitro in a laboratory. And after genetic modification, you replace those cells back into the patient to cure or to treat the disease. In vivo gene therapy, you introduce nucleic acids coated by nanoparticles, genes, whatever genes you want to introduce, you coat by nanoparticles and introduce into the patient, or you clone those genes in a viral vector like adeno-associated virus or adenovirus or lentivirus vector, and those vector you can use to inject into the different organs wherever the functional gene is required. So there are number of monogenic diseases. The monogenic disease is basically caused by a single gene. So if defect in the single gene is causing the disease, we can treat by the gene therapy. So there are number of monogenic diseases like retinal degeneration, progressive retinal atrophy or retinitis pigmentosa, Leber's congenital amaurosis, X-linked retinitis pigmentosa, achromatopsia, hemophilia A and B, leukocyte adhesion deficiency. These are number of different monogenic diseases in animals which can be treated by this gene therapy. And these are the number of studies where successful attempt have been made for gene therapy against the canine monogenic disease in these particular years. Like in 1993, first successful attempt of gene therapy was made for hemophilia B for correcting the factor 9, blood factor 9 through retroviral vector. Likewise, there are in different years for different genes, they have attempted for successfully against the canine diseases using the different genes, functional copy of the different genes using the different vectors like retroviral vector, adenovector, adeno-associated vectors, or fomivirus vectors against the different diseases. Then there is one more area is the induced pluripotent stem cells. It has also got applications in the veterinary medicines. It is basically a type of stem cell that can be generated from somatic cells. Normally, stem cells you can find in the embryo, that is embryonic stem cells. Those stem cells have the ability to differentiate it into any kind of cells. So instead of embryonic stem cells, we can generate the stem cell from somatic cells through reprogramming. That is called as induced pluripotent stem cells. So from somatic cells, we can make iPSCs by transfecting the reprogramming factors like OSKM or OCT4, SOX2, KLF4, NANOG, CMYC, like transcription factors. And then those somatic cells will be de-differentiated into the pluripotent stem cells. And these pluripotent stem cells have the ability to form any cell type of endoderm, ectoderm, or mesoderm. So basically, it can differentiate it into any cell type of the body, like in neuron or intestinal cells. And in the veterinary medicine, this is a promising platform for disease modeling, drug and toxicity testing, biomarker development, and cell-based therapies for regenerative medicine. Especially in regenerative medicine, the large animal represent the best model for man. Instead of doing in mice, if you do in dog or cat or a pig, they will better replicate the process in the human. So they provide valuable system to test the safety as well as potential of iPSC. Also, we develop the therapeutic product in veterinary medicine against the different diseases as well as injuries. Then when we do therapy in the animals, it can provide a very good information if we want to do the same therapy in humans. So it can provide a background information of safety as well as efficacy to plan the experiment in humans. So these are the different areas where we can use iPSC, like disease modeling, transgenic animal generation, human organ generation in animals, gamete or embryo derivation, basic research, biodiversity preservation, biomarker development, drug and toxicity screening, as well as cell-based therapy for regenerative medicine. This is one of the study our lab has carried out in the past. What they have done is develop canine-induced pluripotent stem cells. This is done in collaboration of Anand Veterinary College and Manipal Institute of Academy of Education, Bangalore. So they have developed the canine-induced pluripotent stem cells from the canine skin fibroblast, and they compared with the mouse and human induced pluripotent stem cells, and identified that the canine iPSCs are intermediate of mouse and human iPSCs because it has the marker for both mouse and humans. Some of the markers are present in mouse and some present in the human. One more area which we can explore in veterinary medicine is the recombinant proteins. It is in the limelight for the human medicines also. So examples like follicle stimulating hormones or single domain antibodies, which we can develop in the livestock or in the veterinary medicine. So follicle stimulating hormone is basically used for the IVF. So in the humans, all IVF patients are undergoing treatment by the FSH. So FSH is produced by the pituitary gland. Pituitary gland in the brain, in the adenohypophysis. So in the posterior pituitary gland, this FSH hormone is produced, and through bloodstream it travels to the ovary near the uterus. So in the ovary, it is enlarged view. The oocyte is developed under the influence of FSH. So if we inject the FSH, instead of one ova per month, you can produce more than 10 ova through stimulation by FSH. So in the IVF clinics or even in animal superovulation, we use the FSH for this purpose. So in human, there are a number of biopharmaceuticals are available in the market. Number of companies like Zydus and Intas are making this FSH product they have in the market. But for animal, there is no recombinant FSH available in the market so far in the entire world. So whatever FSH they are producing is from collecting from the slaughterhouse pituitary gland. So we need to develop the FSH hormone through this biosimilar process in the animals too. This is basically a process of the cloning of the gene from the mRNA of the FSH, and FSH has two unit, alpha and beta subunit. So we normally need to clone both alpha and beta subunit and transfer it in the cell line. This is basically people are doing in Chinese hamster ovary cell line. So make a stable cell line and go for the fermentation to produce the FSH in bulk. So from the cell line, you grow in a bulk in a from one flask to multiple flask and roller bottle and then to a fermenter. And in supernatant, this hormone is secreted, which can be again purified by the chromatography. So the another area which we can explore is the single domain antibody from the camel. Because camel, instead of in other species, you will find only conventional antibody. There is two light chain and two heavy chain in the conventional antibody. But in camel, you will find a single domain antibody where there is no light chain, only heavy chain. And in heavy chain also there is absence of CH1, constant region 1 absent. So the antibody domain is single domain and it is very small, around 15 kilodalton. Whereas the conventional antibody is 150 kilodalton. So this camelid single domain antibody are very small and highly soluble and stable in many applications, and this antibody has a potential to treat the diseases like bird flu and Crimean Congo hemorrhagic fever. These are occurred in the animals. So we can produce this camelid heavy chain antibody from a library of camelid antibody by cloning the VHH gene and then screening by differential display technique like fast display or bacterial display technique, and then express this after identifying the gene coding for the antibody, we can express in E. coli and yeast to produce in a bulk and use as a therapy. One more area is the transgenic animal bioreactors, where what we can do is any therapeutic protein like human antithrombin gene. You introduce the gene in the zygote of the animal at the single cell embryo stage, and that the gene will integrate into the pronucleus, male pronucleus of the zygote, and then from that zygote you make the transgenic animal. That's a transgenic animal. For example, if you have cloned the gene below the promoter of milk protein genes, then you will get the protein in the protein production of this antithrombin in the milk of the transgenic animal. So you can harvest the different therapeutic proteins from the milk or serum, or depending on what type of promoters you have used in animals. Even people have done that in plant also. So these are number of examples successfully made these different proteins. These are the different proteins expressed in the rabbit milk using these different promoters of the milk protein genes. And this is the quantity of the protein they have obtained in the milk of the animal. One more area which is important in the animal side is introducing the disease resistance. So disease resistance gene, once we know this particular genes cause introduce the disease resistance, then we can do the generation of the transgenic animal. Like as we have discussed, traditionally people are doing selective breeding. So animals with a desired characteristics are used for the breeding. For example, if one breed of a sheep is resistance against particular nematode, you use that sheep breed for breeding to have the progeny with a resistance in the animal. But that takes a longer time, and you cannot introduce a single gene by the breeding. You get a half genome from the male and half genome from the father and half genome from the mother. But by using the transgenic technology, you can selectively introduce single gene, or you can selectively knock out a single gene which is causing the disease resistance. So there is recently one paper, they have introduced mouse SP110 gene, that is nuclear body protein gene, which imparts the disease resistance against tuberculosis. So they have introduced SP110 gene through tannin nucleus mediated transgenesis in the cattle. And in the cattle they have made by introducing this SP110, they found that they are resistant to tuberculosis. So like this, we can identify more genes which can introduce the disease resistance and make the transgenic animal to remove the susceptibility against many different diseases in the animals. So these are broad areas I have covered in the today's talk. So if there are any questions, we can further discuss.
Thank you, Doctor, for the elaborate lecture on different platforms to develop veterinary medicine. Students, this session is open for discussion. If you have any clarifications or doubts, you can ask Doctor. Any questions? Okay, some question from chat box. Sir, are the single domain antibody different from monoclonal antibodies?
Yes, the single domain antibodies are different from the monoclonal antibodies. Monoclonal antibodies which are made against the single epitope, but they are made up of two heavy chain and two light chain. Whereas single domain antibody is only the variable heavy chain domain antibody. So it is very different, unique and different from the monoclonal antibody.
Any other questions?
There is one. So multi-epitope vaccine is basically you combine the epitope B cell and T cell epitope against the your target pathogen, and those B cell and T cell epitope you combine in a one vector. You can take the epitope against the multiple gene. For example, if you are doing a Covishield vaccine, it has only spike protein gene. But in multi-epitope vaccine, you can do spike protein as well as matrix protein as well as E protein, and you combine this epitope from all these protein and integrate into one cassette to make a vaccine against broad epitopes.
Next question, anyone?
Hello, sir.
Yes.
Sir, my question is, you told about the IPSCs. Sir, can you tell more about how this uses in different medicinal values?
What is the question? IPSC? How it is?
I mean, how it is used in different medicinal values.
Yeah, so IPSC basically, for example, if you have a retinal degeneration, then what you can do is from induced pluripotent stem cell, you differentiate into a progenitor retinal pigment epithelial cell and transplant that into the retina. So it will regenerate the photoreceptor cells. For example, if you have injury into a knee joint and your cartilage has got degenerated, you transplant the induced pluripotent stem cell differentiating into a mesenchymal stem cell lineage and transplant in the knee joint, it will regenerate the cartilage. So likewise, you can do the number of applications for the IPSCs.
So it is only based on transplanting the genes.
IPSC is basically a stem cell. IPSC is not, you are not doing transplanting of gene in IPSC. For making IPSC, you are transfecting some proteins. These are OSKM transcription factors. But basically, when we are talking about the transplanting gene, that is gene therapy. So you are introducing foreign gene, a functional copy of gene in a stem cell. That way also you can do that. That is going to be a gene therapy.
Okay, sir. Thank you.
Okay.
One question in the chat box is, sir, is cloning of animals and human beings is possible from the stem cell technology?
Yes, definitely. From stem cell, you take the nucleus and transfer into the oocyte to make the embryo. So in somatic cell nuclear transfer, you took the nucleus of somatic cell. Instead of that, you take nucleus of the stem cell and transfer into the embryo, and that will make a cloned animal. Or you can directly introduce stem cell into the embryo. That stem cell will give rise to multiple cell lineages, and you will get chimeric animal. And in that case, you have to do couple of generation breeding to get the clone animal.
Any more questions? Okay, no questions. Thank you, Dr. Amrutla, for your wonderful giving us your valuable time today and sharing your knowledge with the students and taking the questions. Thank you so much.
Thank you, sir, for giving the opportunity to present my thoughts.
Thank you, sir.
Thank you, sir.
So Dr. Sandeep.
Hi, good morning.
Good morning, doctor.
Can I share my presentation?
Yes, doctor. So, students, now we'll start our second session for the day. Today we have with us Dr. Sandeep Goyal, scientist F from National Institute of Animal Biotechnology, Hyderabad. Dr. Sandeep Goyal is a veterinarian with a PhD in reproductive biology from Kyoto University, Japan. He spent 20 years at the Center for Cellular and Molecular Biology, CCMB, Hyderabad, as a scientist and a team leader. There he researched in reproductive biotechnology of domestic and wild animals with a specific emphasis on stem cell biology, animal transgenes, testis biology, spermatogenesis, and cryobiology. He has authored more than 50 scientific papers, book chapters, and research articles. Dr. Goyal was a visiting faculty of Georgetown Medical School, USA, and Kyoto University, Japan. Dr. Goyal is currently working at the National Institute of Animal Biotechnology, Hyderabad, developing mesenchymal stem cell cryobanking for allogenic and autologous transplantation of to aid in the widespread clinical application in livestock and companion animals. With this short introduction, I welcome Dr. Sandeep Goyal to present his lecture on the role of biotechnology in animal production. Welcome, sir. The floor is yours. You can start.
Thank you so much, Dr. Murida. Am I right? Or?
Yes, doctor.
Yes. So, thank you so much for giving me this opportunity. Can you see my screen? No.
Yes, doctor. Yes, yes, doctor.
This is the full screen now.
You can go to the full screen view only.
You can move it. I can move it. You can see it moving.
Your voice is a little bit echoing, doctor.
Okay. Can you see the screen moving?
Yeah. Now better. Yeah, I can see the screen.
Okay. All right. Thank you so much.
And good morning, everybody.
My name is Sandeep Goyal. I am from an institute called National Institute of Animal Biotechnology. I have put up a picture for you here. This is an institute which has been come up very recently in Hyderabad. And there are a lot of talented faculty here who takes master students for training and also PhD students. And we are doing cutting edge research on animal biotechnology. So today I'm going to share a little bit of the my research as well as what I've been doing, and would try to keep the talk to a level of undergraduate level, because it appears to me that many of you are in the undergraduate courses. So the title of my talk is Role of Biotechnology in Animal Production. A previous presenter has actually covered most of the things that I was going to speak about, making my role very easy for you guys. So maybe I can focus on certain specific technology now, and I have touched on my presentation to a great deal, as I believe that the post presentation discussion are more important than the presentation per se. So I will not bombard you with a lot of knowledge, but with certain specific knowledges that I think would be beneficial for you. So I think the previous presenter would have defined biotechnology, but I have just one line, one sentence definition for biotechnology, which I find more appropriate and apt. That's the set of techniques by which living organisms are modified for the benefit of humans and other animals. Okay, so there are several definition of biotechnology, but I really like this one. So as you all know, that biotechnology basically is the culmination of or amalgamation of several other techniques and biological sciences, engineering sciences, such as genetics, biochemistry, food science, food technology, mechanical engineering, biochemical engineering, electronics, and microbiology. So it is just not just
Sorry,
a single
doctor.
science. Yeah.
Your voice is not.
Sorry, just a minute.
The mic. Can you adjust your mic? Yeah.
Can you hear me now?
Yeah, now better, doctor. Yes.
Okay, maybe my mic is, placed in a little odd situation. Okay, so
Now,
I'm okay
now
now?
clear, very clear. Now
Right.
very clear, doctor. Yeah,
Sure.
thank you.
Thank you so much. So as I was telling you that biotechnology is culmination of several biological sciences as well as engineering and chemistry. So they all come together to make a technology, a science called as biotechnology. So biotechnology and animal production, I was thinking of talking in detail about three areas, basically reproductive biotechnology, genome editing technology, and transgenic technology. And it appears to me that the previous presenter has covered the genome editing and transgenic technology at large. So I will focus more on reproductive biotechnology given the time constraint that I have today. Okay. So reproductive biotechnology is very vast in animal production. Okay, and it has several areas where reproductive biotechnology works, and they are artificial insemination, in vitro fertilization, sex sorting of sperm, reproductive cloning, and embryo transfer technology. And these are some of the major technology. There are certain other one like spermatogonium stem cell technology and several more, which I do not like to cover in this presentation today, but I would like to focus on these important technologies that has revolutionized the field of animal biotechnology production. Okay, so let's come to the first technology, the artificial insemination. This technology is actually very old, almost now 40 to 50 years old, and it started in India almost at the same time, and the whole technology revolved around one single technique, is that the cryopreservation. So in around 1940, Dr. Polge he was a Russian scientist who worked in US, developed a technique of cryopreservation of semen. And once the cryopreservation of semen was standardized, this technique revolutionized the entire animal production. Okay, and this is a newspaper cutting that I picked up from the internet, that 70 cows are impregnated every hour through artificial insemination in Madhya Pradesh alone. Okay, and there are several states which use this technology extensively, and this is the technology for which the white revolution came in picture, because this technology actually allowed the dissemination of higher germplasm of male throughout the country. So I'll talk more in detail about this. So artificial insemination is defined as a deliberate introduction of sperm into the female uterus or cervix for the purpose of achieving a pregnancy through in vitro fertilization by means other than sexual intercourse. Okay. So this is a clear definition of artificial insemination, that a sexual intercourse is not required. You can collect the sperm or the semen from the male animal, and then you can put it artificially into the genital tract of the female. So what happens in physiologically, what happens that most mammals mammalian female comes in estrus or heat, or they are sexual receptive, or they ovulate once in a month. Okay, and so is the case in case of cattle or cow. They come in estrus, or they are sexually receptive, or they are in heat every 21 days. So if you see here, this is how a cow shows a kind of a state of sexual receptiveness. And if you see the cyclicity of the cows here, so this is marked by a very small window of around 24 hours where the female is sexually receptive. This stage is known as the estrus, and it is repeated every 21 days if the female is not pregnant. And at the end of the estrus, the ovulation takes place. That is, the ovum is released from the oocyte, which is caught by the infundibulum of the oviduct, and then it travels down all the way from here to the uterus. So what happens if the oocyte and the sperm is available, and the fertilization takes place in the oviduct? Remember, this is very important. Many of the student doesn't know that the fertilization takes place in the oviduct. Okay, and then when the fertilization takes place in the oviduct, the embryo is generated, and you can see that the embryo starts to divide from 2 cell to 4 and 18, 16, 32, and eventually forms a blastocyst. And this blastocyst eventually implants itself into the uterus, and the pregnancy is set in. Okay. So what is the advantage of artificial insemination? So it gives us
sorry to interrupt you, sir. Your slides are not moving.
Oh, I'm sorry. Where are we? Which slide I am in?
It's in the first, sir.
Till first.
Title slide, sir.
That's what I was asking. Sometimes it happens that the slide doesn't.
Yes, Professor, you can reshare.
Okay. Is it moving now? No?
Your second one, it came to second one.
Okay. I'm having this issue in long time, so I was just asking again. Maybe I can just move it like this, right? So it's
Sure,
okay,
Professor. Sure, sir.
right? Okay, yeah.
Yes.
So as I was telling you, so I'm sorry, maybe you did not see many of these slides, So I was talking about the estrus in cow, where she's the cow is sexually receptive, and she comes in estrus for every 21 days, and then there is an ovulation that takes place during this time. And if the sperm is there, it meets the oocyte or the ovum, it undergoes a fertilization, and then eventually the embryo is implanted into the uterus, and eventually the pregnancy is set. So the advantage of artificial insemination is that it is a very great tool of genetic improvement. Now what happens here, that the superior male genetic potential can be evaluated rapidly. So there is a rapid proof of male genetic superiority. See, eventually what happens is that one male can in a natural case can breed with maybe 2 or 3 females in one time. Okay, but in this particular case, what happens that the collective semen can be expanded into several hundred or thousands of straws, and then this particular cryopreserved straw can be used for inseminating maybe hundreds of females in one time. Okay, so that is what it is, that it gives a rapid proof of male genetic superiority. And it is, you do not have to have a male in your dairy farm to have this thing. So you can actually have a frozen semen in your dairy farm, and you can utilize this technology for inseminating the female. Aggression is lost. The danger of male, when it is there in the herd, it usually fights with the female. So that is also taken care of. Disease is reduced because there is no direct contact of the male with the female. So the disease is actually reduced a lot. And semen is also analyzed for several diseases before insemination. So it prevents a lot of diseases. It improves the management in the farm, and it gives a better record keeping. And it's very economical to use artificial in a dairy farm rather than keeping one single bull, because one single bull, if it has some issue or genetic problem, it may not be able to so it will pass it on to the next generation continuously. So what happens, that this gives you a lot of advantage to use artificial insemination. So there are several steps of artificial insemination. First is the semen collection, evaluation of semen, extension and dilution of semen, because one ejaculate of a animal of a bull contains almost 1 billion sperm, and that is too high for us insemination. So it can be, a normal insemination required maybe 1 to 2 million sperm, and therefore you can extend and dilute this semen and can be used for several females to be inseminated. And then you have freezing and extended semen. This is a very important part. Freezing is the key where this technology has actually been executed in the field. And insemination of the cow in estrus or heat. So detection of cow in right stage of cyclicity is very important for insemination. So this is a picture of a cartoon showing how it is done. So you take the best bull or the prized bull, using an artificial vagina. You collect the semen from the bull, and then the semen is collected. It is evaluated, extended, and frozen into liquid nitrogen. So liquid nitrogen has a temperature of minus 197 degrees centigrade, and these semen is just filled into these straws. These are known as the semen straw, which is having a capacity of 0.25 ml or 0.5 ml. And this looks like this, and you just fill in this by a machine. This is an automated machine system. The diluted semen is then is injected into this machine, and one side is sealed. Okay, and then this is transferred into a liquid nitrogen container. Okay, after a cryopreservation. So there is a multiple step cryopreservation, and then they are stored in this liquid nitrogen container in these canister like thing. And you can see here that the bull entire genetic, the reproductive potential is now stored in this liquid nitrogen container. So when there is a requirement of a sperm or the semen in the farm, what happens, that you can take out that one single straw from the liquid nitrogen, thaw it at 35 degrees centigrade water bath, cut the one end of the straw, and then you load it into an artificial insemination gun. There is a specialized gun here. It just looks like this. It has have a plastic sheet over it. So you load the straw in between here and put the plastic sheet over it, and it is ready for insemination. It is just like an injection gun. Okay. And this is how it is done. Then you detect the cow in estrus, and you pass the AI gun through the vagina into the cervix, and then one hand is inserted into the rectum, and then you guide the AI gun into the uterine, sorry, into the vagina, and pass it through the cervix. So it's very important to pass the AI gun into the cervix, and at the anterior part of the cervix, the semen is deposited, and then you withdraw the semen gun. Okay, and then you wait for the pregnancy to take place. And after 21 days, if the cow doesn't come in heat again, and within 1.5 days, a very trained veterinarian through per rectal palpation can tell whether the cow is pregnant or not. So this is a technology which has been applied extensively in the dairy industry and at the grassroots level. So this is something which you should know. Another technology of reproductive biology is in vitro fertilization. And as you know, in vitro means the process of creating an embryo or from oocyte by fertilizing them with the semen in a petri dish that is outside the body. The oocytes are first collected from the ovaries of donor by ultrasound guided follicular aspiration, and they are matured in the petri dish for fertilization for 20 to 48 hours later, and then it is incubated for several days for the embryo to develop. So this is the cartoon showing how it is done. So in general, what you take an elite donor cow, you collect the oocyte from it, and you already have the semen from an elite bull. As I told you previously, you can collect it by the artificial vagina, or you have frozen semen, and you subject them to in vitro fertilization in a petri dish, and then you allow them to divide further to generate a blastocyst, and then you can either freeze it, cryopreserve it, or you can do an embryo transfer so that you can have the calf coming out from the elite cow and the germplasm of the elite cow and the elite bull. And the oocyte is collected through this process known as OPU, or ovum pickup, that is done through a ultrasound guided probe, which is inserted into the vagina of the cattle, and there is a long needle attached to it which comes out at this end. Okay, and with another arm in the rectum, you guide the ovary, and then this needle is connected to a aspiration pump which collects the oocyte into a container, and then you actually collect through ultrasound method the oocyte through this follicular aspiration, and this aspirated oocytes are then subjected to IVF through the in the petri dish, and then they're allowed to develop to a later stage embryos, and then they're either cryopreserve or embryo transfer. There's another picture which shows a little bit more in detail that you do an ovum pickup, and then you do an in vitro maturation for 12 to 24 hours. You subject them to IVF, in vitro fertilization, and then you culture them to a later stage embryos. And then from these embryos, you can either freeze them or take a small piece of the embryo cells for biopsies for genomic selection. Okay, so you can actually do a genome analysis of the cells to decide which is a genetic superior embryo. And once you know that which one is the genetic superior one, you can do an embryo transfer into a synchronized female, which is and then you can get the live animal coming out. NIAB has recently released a DNA chip, a genomic chip called as IndiGow, the first cattle genome chip for the Indian cattle for the conservation of pure variety of indigenous cattle breed, which was given to the nation by the Ministry of Science and Technology, the minister himself, Dr. Jitendra Singh. So in vitro fertilization also allows you to do genome editing, as the previous speaker has talked about. So you can actually collect the embryo and subject them to in vitro fertilization, collect the oocyte, subject it to in vitro fertilization, and can do a genome editing at this stage. You can subject them to all these available genome editing tools such as CRISPR, Cas9 or Talon, or and then you can, once you analyze these embryos for genome editing, they can be cultured, and then embryo biopsies can be taken, which can be used for sequencing for their genome editing, precise genome editing in the genome, and then embryo transfer is done to have a genetically superior livestock. Now, coming to my another technology which I'm going to talk about is sex sorting of sperm. So as you know that there is a 50-50% chance of getting a male and a female post fertilization. It is the male who decides the sex of the female, not the female. Okay, because all the time the female egg is always, the egg is always X bearing, but the spermatozoas are the one which bear either the X or the Y. So if we are able to differentiate or sort the X and the Y bearing sperm, it is possible to produce a animal which is either male or a female using the X or the Y bearing the spermatozoa. So in the dairy farm or in the dairy industry, a female is preferred because they are able to produce milk, right? So what happens that there is a technique called as fluorescence assisted cell sorting that can use to sort out sperm based on the difference in the DNA content. As you know, that the Y-bearing sperm has a little less DNA than the X-bearing sperm. You can see here that the testes produce X and Y-bearing sperm. And you can see that the X bearing sperm has more DNA than the Y bearing sperm. So what the technique uses is that they add a vital dye which binds to the DNA. And you can see here that the DNA binding of this dye to the X and the Y bearing sperms are very different. You can see the X bearing sperm has more fluorescent dye bind to it, and when they are subjected to the laser, you can see here that the emission of the fluorescence is much higher from the X bearing sperm than the Y bearing sperm. And this technique actually utilize a computer based technique where it offers a charge to the positive or the negative positive or negative charge to the X and the Y bearing sperm based on the intensity of the fluorescence they emit, and on that basis they can be separated into Y or the X bearing sperm. And the dead sperm which does not get any charge because they do not bind to this vital dye, only binds to the live cells, they do not bind to the dead cells. So then you can separate out the dead sperm also. Now coming to another technology which is very important, and it was like it was really very much in picture in 1997 when the Dolly the sheep was produced. But in our own country now, NDRI and CBRI and National Dairy Research Institute and Central Institute of Research on Buffalo has started to use this technology for cloning the buffaloes. Okay, they've started cloning the elite buffaloes. So by definition, cloning is the process of producing similar population of genetically identical individual through asexual means of reproduction without the use of sperm. Okay, so there are a lot of advantages of cloning. It can use for restored endangered species and conserve the prized animal. Since you have one prized buffalo bull, you can keep replicating it and make a carbon copy of it. It's allowed for the editing of gene in farm animals. Okay, and it is an alternative method for generating transgenic animal as a bioreactors or biofarming. So I'm not going to talk more about that because previous speaker has already spoken about it. So I will talk more about its technique, how it is done. So generally speaking, if you take a single cell embryo, after fertilization, and when it becomes 2 cell, you can actually split these 2 blastomere into individual and then allow them to grow. So that basically means that embryo splitting is the most ancient method of cloning, which was utilized by early researchers like John Gordon and all, in case of frogs and all that. So it was very easy to do this. But in mammalian system, it is more demanding, where you need to use it a micro manipulator system, where you have to take out these embryo under the microscope, one of the blastomere, and put it into a single into a zona pellucida, which is actually used for protective covering over the embryo until it hatches, and then you can allow. So basically what happens, that these 2 embryos generated from single embryo are identical clone of each other. Another method of doing is blastocyst bisection. Blastocyst is the later stage of embryonic development. So what you can do is, here you can take the blastocyst, cut open the zona pellucida, bisect the blastocysts into two half, and then you can actually use one half and put it into one empty zona pellucida, and then when you allow this to grow, they are identical to each other. Okay. What happened when the dolly was produced was a technique through nuclear transfer. So as you know that the most genetic material is restricted to the nucleus of the cell, there are certain DNA which is also present in the mitochondria, and that is the cause of a lot of epigenetic complex that complication that takes place during the embryo transfer. But we are not going to talk more in detail about. So embryo transfer involves a technique where you enucleate a oocyte, you remove the nucleus of the oocyte and take the nucleus from a single cell, and then you can replace this nucleus into the oocyte and then stimulate it to grow, and then you can produce a embryo from it, which can eventually produce a live offspring. So this is known as nuclear transfer technique. Okay. So there are two types of nuclear transfer technique. It is embryonic cell nuclear transfer, where you use an embryo, like for example, this is 8-cell embryo. You remove the nucleus of one of the blastomere, and you take the oocyte, you enucleate the oocyte and remove the entire nucleus of the oocyte and replace it from the nucleus of an embryo. Okay, one single embryo, one single blastomere, and then stimulate it to grow. So embryonic stem cell nuclear transfer has been very successful because embryonic cells are rather less epigenetic reprogram, so they have an ability to reprogram the entire embryo into an individual. However, somatic cell nuclear transfer, which was is another technique where somatic cell is used. Somatic cell is any cell of a living organism other than the reproductive cell or the embryonic cell. So from there, it is a terminally differentiated cell, and if you collect the nucleus from a somatic cell and replace it of an enucleated oocyte, this is known as somatic cell nuclear transfer. But so dolly was produced from somatic cell nuclear transfer, but eventually later research they found that the mammary gland epithelial cell had some embryonic origin also. So therefore this technology has not been very successful in producing a live offspring. Then you have therapeutic cloning. Another technique which is used is that you simply produce an embryo, and then you don't allow it to develop into full term. You just use the embryonic stem cell from the cloned embryo. So what happened here, you can derive a patient specific stem cell. Okay, for example, if I am suffering from some disease and I need my own stem cell, I can take the embryo of certain other origin and replace the nucleus of my skin cell into the embryo or the oocyte, and the stem cell or the embryo generated will be of my origin, from my genotype. Okay, so that I can use it for my own cell therapy. So that gives you a patient specific stem cell. Okay. So now somatic cell nuclear transfer has been used extensively for producing transgenic animal, for gene targeted animal, and also for genome editing. Now the recent technique of zinc finger nuclease, TALEN and CRISPR has been utilized for producing several, and that technique is highly efficient. Earlier the technique of producing gene targeted livestock was only less than 1 in 10 in 1000, or 10,000. Now this technique is almost 25 to 50% efficient in genome editing the transgenic or the animals. Okay. Now coming to the final technique that I'm going to talk about is embryo transfer technology. So as you know that all the embryos produced by the technique of IVF or somatic cell has to be implanted or transplanted into a female. Okay, for it to complete the full term to produce a live offspring. So embryo transfer is the process by which an embryo is collected from a donor female and then transferred into a recipient female when the embryo completes its development. So by the use of embryo transfer, a genetic superior female, please, this is very important. Artificial insemination was for genetically superior male propagation, and embryo transfer technology is for the propagation of a genetically superior female, produces more offspring than she could from the natural reproduction. Okay, so if you see here, this is a picture of a donor cow here, and she was, in a general term, in over a lifetime, a cow would produce maybe at the maximum 10 to 12 offspring. But this donor cow has produced 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 offspring in one time, at one time, okay, in a 1 year. Okay, so you can see here that this technology actually, you know, harnesses the power of hormonal stimulation and can, you know, enhance the genetic potential of a genetically superior female. Okay, so the advantage is to increase the number of offspring sired from a superior female, result in a faster genetic progress, obtain offspring from older or injured animal. So if an animal which is already old and injured, but she is still, you know, genetically superior, you can use this technology. It increases farm income through embryo sales. You can actually sell the excessive embryo to generate revenue, and exportation or importation of embryos easier with the live animal. So these embryos can be actually shipped to another country or in another continent, so that, you know, you can actually genetically propagate that female. So steps of embryo transfer are very simple. You superovulate a donor cow. First you identify a genetically superior donor cow, you superovulate. Superovulation is a process of enhancing the ovulation capacity of a female. So you'd give some hormone like FSH and LH, which actually enhances the number of ovulation in a female. Then you artificially inseminate the female after like 5 days after superovulation, and you recover the embryos from non-surgical method by flushing the uterus, and then you evaluate the embryo under the microscope, and then you store the embryos in liquid nitrogen, and then you would do an embryo transfer to produce a live offspring. So this is how it is done. Superovulate the. So if you see here, this is a superovulated ovary, and you can see here that in a normal cyclicity, only one oocyte is produced by the female in an ovary. And you can see here that there are number of ovulation here, oocyte here, 1, 2, 3, 4, 5, 6. So in a time you can have 10 to 12 oocyte being produced by superovulation. You can see here, this is post ovulation, is the corpus luteum here. Then once the superovulation is done, you inseminate the cow with a genetically superior male semen, and then you collect after 7 to 8 days, 2 to 3 days after insemination, and then you can collect these by, you know, flushing the uterus with the saline or a PBS, and then you can collect the embryos here. These collected embryos are then evaluated under microscope for the quality. So these are high grade embryos, grade A embryos still here, and these are poor grade embryos. So highly the good quality embryos are then transferred into the recipient female. So recipient females are then synchronized with the donor to have the same uterine environment as the donor, and then you do an embryo transfer through the transvaginal letter, similar to the embryo artificial insemination, except that you transfer the embryo more deeper into the uterus. Okay, and this is the result of an embryo transfer. You can see here, this is a donor cow who has produced so many live offspring within one shot of superovulation. Okay, so to summarize my talk, I will end here. The biotechnology has revolutionized animal production by enhancing the reproductive and genetic capabilities of animals. Several biotechnological innovations are in pipeline that are going to further enhance the reproduction capacity of animals. There is a need of fresh talents to continue this innovation in this field. So we need good students and good, talented students to come and join us, so that, we can propagate and progress more further. And ethical issues pertaining to these technologies should always be kept in mind. So there has to be ethical clearance, or ethically everything should be, upright before you do all these technologies. All right. So thank you very much for your attention, and I'm now open for the questions from everybody.
The session is open for questions. Students, first question.
Okay, so I can read here, by cryopreservation, yes, the semen, for how much time period we can preserve the semen, because the life of. Okay, now this is a very good question, and there has been reports of cryopreserving the semen up till 40 years. Okay, even after 40 years of cryopreservation, if liquid nitrogen is maintained in the container regularly, because whatever liquid nitrogen is lost slowly from the container, it is maintained in that minus 196 degrees. Reports are there that up till 40 to 50 years, the sperm has been preserved. Okay, so once the cryopreservation takes place, the cell goes into a inert state. Okay, they do not have any metabolic activity, and when they are thawed, they revive back themselves. Okay, so that is the power of cryopreservation. Okay, any more question?
Any more questions? You can also use the chat box.
So that's why I cut short my talk so that I can have more questions from you guys, because I think, I don't want to bombard you with too much information. So we actually conduct a summer training program in a institute. So sir, does Siemens Sorting has any, see, ethical issues are there always. Ethical issues cannot be, ethics and science goes together. Okay, but when the advantage of certain things is for a human benefit and for a larger picture, it is always given a preferences. Okay, so Siemens Sorting obviously do have ethical issue because you are actually trying to get more females from than the males, right? Or vice versa is also possible. Even in human, it can actually have another implication, but it is ethically wrong to do a human semen sorting. Okay, but in case of livestock, in cattle, it is ethically all right to do it, because the farmer needs more females in her farm, so, and the males are actually slaughtered in this country. To avoid that slaughtering ethically, so you can see here the weight of advantage is much more than the ethical concern. So obviously it is all right to use semen sorting in livestock.
Any other questions?
That's possible, say, losing a superior male, basically what happens that in a dairy farm or usually there is only one or two males to inseminate most number of females. So there are now technology through which we can actually know the superiority, genetic superiority of a male, so the possibility is there, but the possibility is not that high. Okay, definitely when you're subjecting more female, you are losing the other sex and giving an advantage to another sex, right? But the possibility of doing that in farm setting is quite low.
Doctor, you are about to say some training programs, right? Conducted.
Yeah. So what happens that in our institute every year, twice a year, we have a training program for MSc students or BTech student who can apply through a proper channel. There is an advertisement, and we actually allow them to come here for 6 months and work with a given scientist, depending on the interest of the student, what they want to do, and then they they contact them. So I welcome you all, whoever is there, to apply for these programs so that you can get a good hands-on training on these modern area of biotechnology.
Students, you can check the NIAB website. Maybe you will get the links, all the details there, and
Yes.
interested persons can apply. If no more questions, we will wind up this session. I thank Dr.
I think BTech students are also eligible. Yes.
BTech student. So I thank Dr. Sandeep Goyal for joining us today and sharing his valuable knowledge and experiences with us.
Thank you so
Thank you
much.
so much, Doctor.
Thank you so much for giving me the opportunity to connect with the bright minds of this country who are the future for these biotechnological intervention and innovation. So thank you so much.
Thank you so much, Doctor. Good morning, Dr. Binod. Dr. Binod Kumar? Professor? Professor Binod? Yes. Good morning, Professor.
Good morning. Good morning.
Good morning. Students, we'll start our third lecture for the day. Today we have with us Professor Binod Kumar. He is a professor and the head of the Department of Veterinary Parasitology, Veterinary College, Kamadhenu University, Gujarat. So Professor had a Bachelor in Veterinary Science and Animal Husbandry from Veterinary College, Hyderabad, and a Master in Veterinary Parasitology from ICAR IVRI, Izzat Nagar, and her PhD from Veterinary Parasitology from Izzat Nagar in 2006. So joined as Assistant Professor to the Veterinary College, Junagadh, Gujarat, presently working as Assistant Professor, Head of the Department. His major research areas are anti-tick vaccine development, tick-borne disease diagnosis and control, tick control, and wildlife parasites. He has an expertise in recombinant DNA technology, RNAi technology, molecular parasitology, acaricide resistance, PCR technology, lab assay, herbal drugs, parasite vaccine study. He has published more than 20 papers in international journals and 30 research papers in national level reputed journals. He has 10 popular articles, 4 books, and 5 book chapters, and has 5 training and practice manual. He had several awards to its credit, like ICAR Senior Research Fellowship, ICAR Net Qualified, and Dr. J.P. Dubey Young Scientist Award by IAAVP in 2010, 3 Best Poster and Oral Presentation Award at various conferences and symposiums. So with this short introduction, I invite Professor Binod Kumar to give his lecture on herbal treatment for ectoparasites in cattle. Professor, you can start.
Okay, thank you, sir, for introducing me and giving the opportunity to interact with the students from all over the countries and to interact and to give the information on the parasites, especially ectoparasites of the cattle, and remedy to control it or to rid of it. One request from the host that my internet connection is password protected, and in between they automatically logged out. So when it is logged out, I will not know that it is logged out or what. So you have to intimate me that your voice is not coming. Then again I have to log in.
Okay,
so every 15-20 minutes it automatically logged out. So I have to log in again. Then only I will connect it.
Okay.
So please that. So shall I start my presentation now? So good morning to everyone, dear participants. I'm going to discuss about the herbal treatment for ectoparasites in cattle. This subject is very vast, and I try to give the informations that give you the initial introduction that what is the ectoparasites and what are the research and development going on regarding the herbal treatment, and what are the some things used in the field for a control of these ectoparasites. So I have divided my lecture like that. That first of all, we should know what is the parasite, ectoparasite, then what are the different kinds of the ectoparasite affecting the cattle. It is its economic importance, because knowing the importance, controlling is having no meaning. So we should know that what's important, then only we will think more and more that it should be controlled. Then it's a resistance and susceptible life stages to the drugs, so that we have the idea to which stage to be targeted. Then control methods, what nowadays practices are going on, what control methods are available. Then what's the ethno-veterinary practices in short, and then we'll go through the herbal formulations, research and developments, and then finally challenges and limitations. So we have 45 minutes of the lecture. I'll try to complete this all the topics through the discussion, and then finally we have the question answer session. So that now we should know first that what is ectoparasites. So these are the ectoparasites. I think many of the parasite is well known for everyone, but few are the parasites may not be known to the majority of the participants. So this called as a fly. They are broadly called as a fly or a lice or the fleas. They are the insect. They are classified under the insect. And the lower panel we can see ticks and the mites. They are called as acarines. So these five class of the things, or broadly we can say this fly, lice, flea, ticks and mites. So this 5 different class of these arthropods mainly affect the cattle, or we can say animals. So this again have the many different species or a genus, and having the different kinds of their affections to the animals. Okay, so these parasites affect the external surface of the animal. That's what we say the ectoparasites. So this first we go through the fly. You can see here many kinds of the flies there. These all kinds of the fly affecting the animals. They are of different kinds, and they are classified based upon certain morphological features and their behaviors. Some are called nematoceran fly, like you can see in the upper corner right side, this left side, this fly of different kinds, where the mosquito are there, black, and some small fly that is called culicatel. They are very common fly in the tropical countries, and they are biting fly. They are simply biting, and then they require the water for their development, their breeding. Okay, so they have the this kind of habitat. Then next is that next corner is the this called the cycloraphan fly, and that that brachyceran fly, and that brachyceran fly is also biting fly. They are blood sucker. Their severe painful bite is there. Okay, and they also require the water or moisture for their growth and development. Their breeding places, this moistened place or the muddy area or the watery area. So they again require the water. Next is the in the lower panel you can see one side 3 flies there. That is again biting fly. Lower panel fly it comes under cycloraphan fly, and this is again biting fly. Here you can find that many different kinds of flies there. Their importance are different. Okay, so this biting fly, some are biting fly, they are blood sucker. Some are non-biting but very good to transmit the diseases, and others are they are non-biting but their larval stage, means their immature stage are important. If you know that the different life stages, because lower group of organism they are having the different life stages, they are different in looks. Okay, their morphology are very much different, and you can see for the fly lay the eggs, then have the larvae, and then pupa and the adult fly. Okay, so these 4 stages is there in the fly populations. And two, adult and the larvae is feeding stage. They feed, they had the mouth. Eggs and the pupa is non-feeding stage. So here in the some of the fly, which you can see a colorful, very much colorful, their larvae is important. Their larvae cause the disease in animals. The adult fly is simply feed anywhere, any decaying organic matter, any dirty things they are on that. But their larvae, when they comes to the animals, they give the larvae, and larvae goes inside the body. They infest the skin or their internal organs of the animals. Okay, so here many kinds of the fly is there, which called the masses causing fly, because the larval infestation is known as the masses. So here now we understand the what's the life stages of the fly, what are the different kinds of the fly which affect the animals. Okay. Then another group of the insect are called the lice. I think everyone know this lice is very common in human too, but in animals also they are very common, and they cause the some kind of the irritations leads to the different kinds of the problems in animals. Here their life stage is somewhat different, and this is the permanent parasite. One thing we should know that their habitat for the fly, their habitat are for the different fly, they are different. Hello. Hello. Yes. So this here this we can see that this is the here the habitat is important. We find that they have the both. We have the this both kind of the development. Some stage on the animal, some stage on the environment. Here, in case of lice, all stage on the animals only. They don't goes to the environment. Their development entirely on the animals. They are called permanent parasites. Here, another side we can see the pupal stage, where you can find that their life stages are both on animals as well as on the of the host, means in environment, in the animal houses. So that here again similar to next parasites are the ticks. Here many stages are there. And this is very problematic one in animals. We can find that they have the larvae, nymphs, adult, and again have the many stages within the larvae and the nymphs. And their development are according to their species. They are called one host ticks or two host ticks or three host ticks. So this biology also very much required for the management of these arthropods, because we should know that which parasite, which stage of parasite, where they are, what is their location, and then only we can have some strategy. Okay. Then mites, it's a totally permanent parasite. All stages on animals only. Nothing will go on the surface or ground or animal house. When the parasite comes on animals, they lay the eggs in the animals, their development on the animals, and they cause the disease accordingly. And majority of time you will find this is very much microscopic. They are microscopic. You need not to see by without any microscope, unaided eye. Okay, so that's what this is the very unique arthropods among all others. Okay, so this. Now we understand the different kinds of arthropods, their habitat, their locations on animals as well as on the environment. Okay, we should know that's what's the importance. Why we'll go for the this control of this parasite? Why control is required? Many arthropods are there in environment. We don't require, but here these parasites are very important because they have the both direct and indirect impact on animals. Direct, they are very much biting. Their larvae cause the disease in animals. They create the annoyance in the animals, and this way leads to the reduction in the production. They severely affect the normal physiology of animals. And secondarily, most important is they are very good biological vectors. They transmit number of the pathogens, which may be bacteria, virus, protozoans, or rickettsials, or even helminths. Okay, so they transmit variety of the pathogens, and they are very serious. They produce the very serious disease. That's what we should have some strategy to control it. Then the expenditure on control is also one of the costs against this. Okay, so that is the things. If you see the in the term of monetary terms or in the measurement terms, then we find that some of the parasite assessment is there, estimates are there, where we can see that these arthropods, they cause the huge loss, economic loss. We don't find that they directly cause the death, but they have the slow action, and they, because of transmission of the pathogens or because of their direct impact as irritation, annoyance, and that this restlessness of the animals, and they lose their energy in that, and that their energy are not utilized for their production, and we find that there is a loss, huge loss to the farmers economic terms. Again, ticks, among the all arthropod, ticks are very much important, especially in the cattle. Nowadays, every farmers, if you go to the farmers and if you ask them that what is the problems, they will say the tick is the major problems. They are not able to control it, and this is the problems because we don't know the actual the control strategy what to be taken, and other problems is the use of the drugs. That's, we will discuss these things also. So this ticks, again, you can see it's a worldwide problems, and India is not away from these problems, and you can find that that's leads to the lot of losses to the farmers in terms of the production loss, in terms of the disease transmission, in terms of the control measures. Okay, so that all leads to the very high losses to the animals. Now, for the drug development perspectives, we should know what is the susceptible stage of the insect or arthropods. Okay, so what are the susceptible stage? Which stage of arthropods should be targeted for the control of these ectoparasites? So in the initial slides, we have seen that for the fly, some stages are there, like the adult, larvae, eggs, and the pupa. For the flea, similarly, it is there, like the fly. Lice, it's having the somewhat different, that it's having the eggs, larvae, and the this or not larvae, eggs, nymphs, and the adult stage. So this for the insect, we can see that the many stages are there, but all stages are not susceptible. If you put the drugs, any kinds of drugs on the eggs, it will not affect the eggs because they have the very protective layers, and the inside the what organism, the embryo is there, developing stages are there, they are not affected by the external applications. Okay, so what stage which is susceptible to the drugs? So the stage like the adult fly as well as their immature larval stage or nymphal stage, which is there either on the animal body or in the set. So mostly you will see for the fly, larvae you can find in the water in case of nematoceran or brachyceran fly, like a mosquito or a tavanus or like that fly, or you can find the organic matter like the Stomopsis, musca, different kinds of these hippobosca and other flies, biting fly. And if you see the masses causing fly, their larvae develop into the animal body, they are susceptible. So there you can see that habitat of that susceptible stage, knowledge is also important. Then only you will think that which stage to be targeted, and target stage is where it is, what is the location, and accordingly the drug should be developed. Okay, then flea, again you will find larvae of flea is on animal set, their larvae is susceptible. Nymph of the lice on the animal body. In adult stage, in case of insect, we can find that they are either on the animals or either in the animal houses or organic matter. For the flea, animal body, and lice also. So for the insect, we have to target either the animals stage or of the animal stage, which are there at different locations, different organic matters or the waters or this animal set. Okay, so that is the things. In case of occurrence, somewhat different issue are there. Okay, because here many stages are there, and according to the host of the tick, we know that Rhipicephalus microplus, which is one host ticks, and which is very common. More than 900 tick species are known in the worldwide, and in India 106 species recorded till now. Okay, reported by Ghosh et al. 2006, and among that 5-6 species of ticks are very common. Okay, in that Rhipicephalus microplus is most common. Rhipicephalus microplus, they are known as the tropical cattle tick, and another is Hyaloma anatolicum. So these two ticks are very common here in the Gujarat also, and in overall India you will find that these two tick species are very common in India. Hyaloma anatolicum is 3 host ticks and Rhipicephalus microplus is 2 host ticks. So knowledge about this host is also required, because we know that from that the which stage is on the ground, which stage on the animal, and accordingly our strategy, our drug development should be there. Okay, another things is mites. Mites are always on the skin only. You will not find mites on this animal house or surface of the earth. Okay, so whatever drugs should be developed, that will be taken with that strategy, that it should penetrate the skin, it goes through the skin, it will affect that the particular animals. Okay, this particular parasites. So these are the different susceptible stage of the parasite that should be targeted, and they can be targeted, and accordingly the drugs should be developed. Now, what are the control methods available and till now? Okay, so they are classified as the chemical control methods. They are called the chemical acaricides, or insecticides. Biological control methods are available, like some biological materials, maybe some arthropods, they are called hyperparasite, or some another nematodes, they are called the nematodes. Then have the some these birds also used for the control of the arthropods. Some fishes are used. Okay, so they are biological control. Immunological control means some vaccines are also available, like the tick vaccines, like tick guard and gavak, but they are located to a certain region only because of some disadvantage. Yes, phytoformulation, which we are going to talk about that. Then genetically resistance breed can be taken, like we know that Indian breeds are somewhat resistant than the exotic breed or crossbreed. Biopesticides. So in that all, if you see the use wise or a development wise, the chemical acaricide is mostly used, more than 90%. The present strategy is based upon the chemical control only. Now we want to shift to the phyto. Why we want to shift to the phyto? Because biological controls, immunological controls, genetically resistant breeds and biopesticides having their own problems, limitations. Though they are very eco-friendly, very good, but still they are many are, we don't have the these things to be used as a wider scale and in the field. That's what we can find, that the another control method, that is phytoformulations, may be the answer of the chemical controls method. So in generally we find that chemical controls are very high in used, and because they are very much target specific, their mode of actions are very much defined, and they are act on that, and that's what you can find, that they are working very nicely, and we can see after application that arthropods are dying or it is controlled.
प्रोफेसर आपका कनेक्शन लॉस हो गया, वो टाइम खत्म हो गया था ना। Professor, we lost your connection. Professor?
Sorry, sir, you are muted, sir.
It is unmuted. Yeah.
Yes, Professor.
Yes, yes. So these are the drugs which is used presently, but they have the limitations like the development of resistance, contamination environment, because they are acting non-specifically to the target organism. They act on target as well as non-target also, and we find that residues, milk and other animal products, and that's lead to the many problems. So by looking that, again we are shifting towards the ethnoveterinary practices. So reviewed are there, many review are there where they review that the many of the old drugs which people have the knowledge to control the parasites or control the different kinds of the diseases are used earlier. Even our this old literatures having that all kinds of the things. The problems is that this ethnoveterinary practices that it is inconvenient to prepare and to be used, though they are some are very good and known that that is the effect is there, but the problems are there. Certain problems are there, like availability of plant is seasonal. So seasonal plant is there. So tick problem is there in the summer, and that time we don't have that kind of plant. That is the problems. Sometimes we will find that the dose are not standardized. Those ethnoveterinary is there. So many times we find they have some side effects or they may be harmful because those standardization or their standardization was not there. Just people to people knowledge is transferred, and this way we can find that those are uncertain or they may be harmful and their action sometimes may be very slow. Okay, that's what again research has been taken to develop some defined product from the plants, and that's what now research came for the herbal formulations. We know that large number of the plants, more than 45,000 in India, known to have the medicinal value as per the review from the Kumar 96. And even more plants are there which is having that medicinal value. Here, few plants are used by the Ghosh and Ravindran. Actually, I was done my PhD in Dr. Ghosh laboratory, who is working since 15 to 20 years on this herbal acaricides. And I was also associated with that all the persons who are working on with that. Actually, my work was on the vaccine against the arthropods, means tick vaccine. But so my focus was on tick vaccine as well as the laboratory is continuously working on this herbal along with the National Research Botanical Research Institute, Lucknow, as well as Central Drug Institute, CDRI, Lucknow. So we have the this all kind of the this association, and we always have the discussion that what's going on. So these all the plants tested, and then it was found that this is the flow chart. By this way, we can go for the extraction selection of the study of the herbal this product. So what should be the if somebody wants to start the research on the herbal formulations, then they had to go for first of all review of the literature, where the literature mining is required. Lot of things are there, many books are there in our here library. Also we have the 30-40 volumes of this herbal drugs these books. Okay, so lot of things are there on the publications also. So we have to see that which is important for the this ectoparasites and which is available in India. It's not that available in the outside the India and we are working, and that also that they are very common, because we don't find that they are common, and then how we'll work on that. So that is, then we have to select that one. Again, we know that the different parts of the plant having different kinds of the ingredients, their the constituents are varying with their parts. So that also through the review we can know that with parts having the somewhat the efficacy against the some kinds of the arthropods, so that we have to select and then go for the extraction. So extraction may be aqueous or organic phase extraction, and further this extraction can be done for the this identification of different kinds of the extract based upon different organic phase, maybe of hexane or methanolic, okay, acetone. Different phase are used to extract, and with the different extract we get the different kinds of the active principles. Formulations we can say, and then every the extract what is collected goes for the in vitro testing. So in vitro testing, for that again we need to have the some kind of the model for the testing, and where required the biological materials, growth and development of the laboratory rearing of the arthropods, then only we will get the regular that arthropods. In the ticks, it's a very well set up this protocol is there to how to rear the ticks. Okay, so we have this all kind of the experience and that how to rear the ticks, where the lab some instrumentation is required, and as well as animals are required. So both are used together to grow the good number of the arthropods to test it. So after testing, if you find that some of the fractions, some of the extractions, either aqueous or organic phase is having some efficacy, then we go for the chemo profiling. We have to know that what are exactly the different kinds of the chemicals it have, what is the major one. So accordingly, we have to profile with the different kinds of the technology. We have the HPLC, or we have the gas chromatography. With that, this chemo profiling done, then again many fractions are identified, many ingredients identified, active compounds may be identified, and then they again synthesized and go for the in vitro testing and finally in vivo testing. So this is the here continuous process are there. A lot of work is there to extract, to profile, to go for in vitro testing, then finally in vivo testing. So this many a times it was found that when we have the direct crude extract from the either aqueous phase or organic phase, they have the some efficacy, and then we going for the this separation or narrowing down the different components of the fractions, then the efficacy may goes down or maybe have the some kind of the better efficacy. Okay, so both the things are there. So sometimes it's find that, or majority of times we find, because I have working with that, when we go for the chemo profiling and identifying individual things, then the things was very different. That what we have the good efficacy 70-80% or 90% in case of this crude extract. When we go for the purification, then the efficacy reduced. So because in the crude form we do not lot many things are there. Okay, so that is the thing. One of the example here, the profiling of that. So work has been done in the same Dr. Ghosh lab, where they have did the resinous communis used for the fractionization and identification of the extract. So here they have used these leaves of this plant, and they did the serially that fractionation, and initially they were extracted ethanol, and finally they were in the different mobile phase, maybe some extracted in the organic phase, some again insoluble phase, chloroform phase, butanol phase, and every phase having some other kinds of the different kinds of the compounds, maybe of flavonoids, glycosides, alkanoids, tannins. So they are extracted and then they were tested. So we know that from this, that this plant have many plants were tested, and the plants known for the some kind of the efficacy, especially actually I am talking frequently about the acaricide. Actually, acaricide are the drugs against the ticks, used against ticks and mites, because ticks and mites classified under the acarines. And most of the research are there on that ticks and mites only, especially ticks research is going on, because ticks are our major arthropods, which affects a lot to the animals. both direct and indirect way, because many of the parasitic diseases, bacterial, viral, or protozoan parasites are transmitted by these ticks, and we find that this huge problems. So this research is also focused on that. Most of the when I reviewed that, we find the majority of the research is on this acaricide control only, phytoacaricides. Okay, so this some of the important plants known for the this having the good efficacy against the ticks. Okay, so these are also list of the plants known for having the anti-tick activity, and the some of the important ingredients available with that, natural products available with that is listed here. Okay, Anonaspora moja is very known and very old studied parasite. The plants material. So like that here, one list is there where the even location wise they are classified, that which part of the India the plant is available. Because that is also very important, the location and availability of the plant material. So this is also is a very important. Now, mode of action. So we don't know much about the mode of action regarding the this herbal formulation, because it has the many component, many unknown component also. Okay, but some research was done, and some things known about that, like gingiber officinalis inhibit the acetylcholine storage, like our this OP compound they did. Similarly, the essential oils, like they interfere with the GABA gated chloride channels. Sorry. So this receptors, action on receptors of monoterpenoids also evaluated by the events 2005. So very few information are available about the mode of action, because they are not exactly the final compound. They have the many of the ingredients, many of the constituents that that affect the act on the arthropods or this targeted organisms. And accordingly, few commercial products are also available in the market of plant based. So this is the few product I have listed here: Irina, Jerokit, and Neem Prop, Ecotrol, Ceprop. Okay, so these are the these are the used. Actually, these all things. The idea came from this. We know that the pyrethroids. This is the very commonly used nowadays drugs, especially in animals, and in our homes also we used in the day-to-day life synthetic pyrethroids. Okay, they are actually the knowledge came from the plants only, where the chrysanthemum plants, the flower, the initially they were pyrethrin was extracted, and it was known that they are acting on the sodium potassium channel of the arthropods. But they are very much biologically degradable in the light, air, and moisture, and that's what later on they were active principles were identified and synthesized, and later it was found that they are very effective. The problem is that again now we have the problems of resistance, because we have the separated the active ingredients and did the research, synthesized and used it. They had the single target sodium potassium channel. So now it was known that the parasite arthropods now have the resistant population have the somewhat change in the this sodium potassium channel, and this way they now become a resistance to these drugs. So what I mean to say that when we go for the single this active principle, single constituent based control, then there every chance of the development of resistance. That's what now people are proposing to have the formulations where every kinds of the things are there, constituents are there, and they act synergistically, and development of resistance are very minimum, because here the components are many and their mechanism of action may be of many of kind. Okay, so that the things. two important development recently happens. I want to discuss here in that context. The one of the important herbal medications nowadays they are prescribed and people are using. It is under the this institute, the National Innovation Foundation. Actually, National Innovation Foundation is the program run by the DST, funded by the DST, and they developed this one herbal product, that is where they use the neem as well as the monk's pepper, this plant. Okay, their leaves were used. So here protocol is written. This can be found in the ICR website. Okay, so I have given the link below. There you can find that they use this neem and the monk pepper, leaves, and they develop the herbal formulations and used on the animals to control the ticks. I told you the ticks is very severe problem in the right now in the cattle industry. So this was found that they're very effective, and cost of control, which was earlier very high in terms of the chemical drugs, and they had the many other side effects and others. In that context, that the cost of the treatment also drastically reduced, as well as this found that's very effective. And this is tested on the pilot basis in the Haryana state in the few district, and it was found very effective in the field also. Okay, so this can be information can be one can get from the ICR website. Okay, another things, it's developed at the Parvani Veterinary College. Okay, there Dr. Nadlerkar. So they have did the same neem. We all know that neem is very well known since a long time from the ancient, that's having the anti-insecticide, means it's having the pesticide this activity. So this neem kernel they used, and then they have this procedure where they soak this in the water overnight, and then they did all kinds. They use even the detergents or a soft soap, and they found that this spray is very effective for the control of the ticks. So these kind of the things are going on, but this is at the local level can be done. Okay, so local level can be done. Then how it can be used in a commercial scale? That is the problems. So what is the challenge and what's the opportunity? Challenges is that the what formulation we have, it's not called as a true insecticide, because they have the crude extract or some are refined. Still they are not defined under the drug rules, that it is the drugs. They had the slow effect. They are rapidly degraded in the UV sunlight. So very important, because animal, when we apply to the animal will not housed always. They go outside, and at that time the effect of that gone. Okay, no established residue tolerance. Then legal registration is not there. The phytoformulations are not comes under the Drug and Cosmetic Acts, and you will find anyone can prepare anything. Variable efficacy. So that is the challenge. Variable, the efficacy is very much varying. There depends upon the plant to even the plant, same plant from the different geographical locations. If you collect and if you see the efficacy, they are variable. Same plant having the variable efficacy, because the soil is also affect the amount and content of the plant parts what is there. So that's what the standardization of these phytoformulations are very difficult, and that's it's difficult for the regulatory process. Opportunity is that it's a rapid in degradation, so we don't find the residues in the food as well as the environment. So what we have the residues problems in the drugs, chemical acaricides, chemicals, insecticides. No, it's not be here. The effect is narrow spectrum. You can find that they affect only targeted species. In case of the chemical acaricides, they are target or affect both the target as well as non-target organism. Okay, here we develop the phytoformulations for a particular target. They will mainly act against that only. Okay, so then we can find that some are having the repellent as well as medication value, and then you can find that resistance development is very minimum, because here we have the multiple things in a formulations, and then they might have the different kinds of the action and different way of the action, and that's why development of resistance are minimum. Okay, so this is the opportunity and challenge, and with that we can conclude that the phytoformulations are very effective, but the things we should have some standardization, some markers. We should have some markers to assess the compare the compound which is effective against the ectoparasites. We don't have till now any markers to scale that yes, this plant is having this much of the effective concentration of a particular component which is there, which is working against the parasite, which is against the, so that identification of these markers is very much required. Against that standard we can use to standardize any plant material to say, then only the commercial product is having somewhat or can increase the value of that commercial product, and we can have the product, more and more product in the market. So opportunity is there, lot of opportunity is there. Lot of research are going on nowadays on the phytoformulations, and because the drugs, we know that resistance development and this all are very much very high nowadays in the farmers. When we go to the fields, they are saying that what drugs they are using, sapermethrin, deltamethrin, ivermectin, they are not working. They are repeatedly they are using these drugs, and they found that the parasite is not controlled. Ectoparasite are not under control, and the cost of the treatment are increasing day by day. So farmer is under the stress regarding this, especially the tick control. So with this, I think the students, those are the participants, may got the some this knowledge about the what's the development, research and development going on the herbal drugs, as well as what is exactly ectoparasite, what are the targeted stage, where their locations. So with this, the newly graduated students, they can take up the research in that direction and can develop the technology, develop the formulations for the future control of the arthropods. So with that, I conclude my this presentation. If any questions are there, it will be welcomed.
Thank you, Doctor. The session is open for the discussion. Any clarifications you can ask Dr. Binod.
Yeah. So one question is superior males also. What's sir? BTech students are eligible.
That is from the previous.
Yeah. So this. So BTech students means actually this many institutes are there in our here also in the veterinary college. We have the collaboration with the pharmacology department. The pharmacology department having the even the one project on that plan project on this identification of the various the plants, regional plants used for the control or treatment or as a medicinal plant, plant identification and standardization. So they are actually in the veterinary college only veterinary student, BBSc and AS student get the admission for MBSc. Okay, no BTech student, but many other institutes that I told you, that the NBRI, National Research and Botanical Research Institute, and then National Botanical NBRI, and then CDRI, their BTech can get the admission, I think. So there can be done research and all. So general university it can be, and every university, I think herbal, everywhere the research is going on. Anything else? So here also we did the research on herbal as well as the tick control. Here, this region also have a lot of tick problems, and this is the answer phyto only, because in vaccinations the research are very costly, and then the chances of getting the product is minimum. For the phyto formulations, though it's a very cumbersome, very lot of work is required to do selection, screening, and all in vitro testing. Many things are there, but this is have the opportunity. So, any questions? If more question, if it is there.
Yes, good afternoon, sir.
Yes.
Sir, I'm also working on the ticks from the herbal extracts.
Yeah,
So
yeah.
I want to know about what are the techniques, or how can we uh, find some specific markers or components that is for specific plants. What are the components that is showing acrositical activity towards ticks?
Okay, that's actually everyone talking about the marker. Marker means the one standard that in that comparison we can say the some active ingredients are in that concentration, the ratio of that, and then only we can standardize the product, scale up the product. Okay, so that markers still it is debatable that what markers should be taken. So that is not still defined that which plant constituent can be taken as a marker. Okay, research are going on, because if some of the plant constituent which is constant in all the geographical regions in a same kind of the plants, then we can say that constituents can be taken as a markers, because they are consistent. They are not varying with the season. They are not varying with the this geographical location, soil type, and all. Okay, so then only we can say, because the what the active ingredients are there when we identify, they vary with the geographical location, seasonal variation is there. Okay, so how we'll control it? Because when we go for the final production, and that time we will collect the plant from all the season, we will collect the plant from wherever it is there, then we will get the different kinds of the level of the or concentration of that ingredients, active ingredients. So that will not exactly what we will get the in our research and development. So that's what we should identify, and then only will scale up. So that research is going on. Still, it is not indicated that this particular product taken as a marker for standardizing the dose or concentration of the ingredients, active ingredients.
Okay, okay.
Any other
And,
questions? Yeah. Yes, Deepika.
Yeah, please.
Deepika?
Yes, sir. I'm asking there are some very much there are literatures are there regarding this markers and the components which are affecting for acrositical activity towards the ticks. So I want to know about what are the mode of actions ki,
Yeah, mode of action means we know that from the chemical side, the mode of action is maybe they affect the nervous system, they affect their water regulation system, they affect their this metabolism process. So anything, because in the plant formulations, we don't know how many things.
so you are not audible. Maybe.
Doctor? Doctor? I think we lost you, doctor.
Yeah, I have lost the connection. Now again connected.
Any other questions, students? If no more questions, we will close this session. We thank Dr. Vinod Kumar for enlightening the students about the ectoparasites and how to control them using herbal drugs treatment. We thank you for sharing your knowledge and expertise, Doctor. Thank you so much.
Thank you, sir.
Thank you.
Thank you
to all for inviting me and giving me opportunity to interact with the students. Once again, thanks to all.
Thank you. Good afternoon, Dr. Kamini.
Yeah,
Ma'am?
yeah. Yeah, good afternoon. Yeah, good afternoon.
Students, we are entering our fourth session for the day and final one. Today we have with us Dr. Kamini. She is a senior principal scientist, Department of Biochemistry and Biotechnology, CSIRI, Central Leather Research Institute, Chennai. Is having experience of 28 years in research and teaching. She has completed her MSc and PhD from Madurai Kamaraj University, Madurai. And postdoctoral experience awarded International Science and Technology Award for STA by the government of Japan for carrying out postdoctoral research work at the National Research Institute of Brewing, Hiroshima, Japan from 1999 to 2001. Her area of research includes microbial enzyme technology, bioprocessing, genetic engineering, waste utilization, production of alternate fuels, and polymer degradation. She has 13 patents to her credit, out of which 4 patents were granted. Technology has been licensed recently to Talco, Dindigul Tanners Environmental Control Systems, Dindigul. She has more than 70 research papers in international journals of high impact value. Her project, she has completed many international joint research projects with various countries like Japan, Bulgaria, Argentina, Belarus, Germany, and national projects from DST, DBT, CSAR, and Ministry of Environmental Forest. Dr. Kamini has received various international awards, including CSAR DAD, JSPS Invitation Fellowship, and IJAA Medal Orientation Award from Government of Japan. She recently received National Award for Technology Innovation under Polymer Waste Management from Ministry of Chemicals and Fertilizers, Government of India. She has guided 6 students for their PhD, and currently she is guiding another 6 students. Apart from this, she has mentored 120 students for their completion of project work in UG, PG, MPhils, and MS by research programs. With this short introduction, I invite Dr. Kamini to deliver her talk.
Yeah, thank you for the introduction. Yeah, thank you very much. Yeah, good afternoon all. Can I share my PowerPoint presentation?
Yes, Doctor. You can.
Yeah. Hope it is visible.
Yes, Doctor.
Good afternoon all. The title given to me was Microbial Enzymes for Industrial Applications. So microbial enzymes are of great importance because of their unique characteristics. They exhibit broad range of pH, temperature, and substrate specificity. We can easily develop modern technologies in place of conventional chemical methods in solving problems in various industries. And the processes, whatever we are developing with microbial enzymes, they will be eco-friendly, and depending on the need, the enzymes could be produced. So development of processes using enzymes are research-based and interdisciplinary, and ultimate goal is commercialization. Always they are non-polluting and help in saving energy. They are less accident prone and easier to train people to handle technologies. And the work, whatever we are carrying out, it will be interesting and exciting for those involved. So considering the microbial enzymes, they could be produced from bacteria, fungi, and yeast. Though there are several microorganisms available in various culture collection centers, always there exists novelty whenever we screen our own microorganism and we can produce some product. So that will be of novel. So screening of novel microorganisms is essential due to the diversity of the organisms in nature. The selected microorganism should be non-pathogenic and generally regarded as safe, and the organism should not produce any toxin, and the product should be produced within short time, and it should be cost effective. So whatever organisms we are selecting it as novel, the process and the products developed are to be patented. Microbial cultures are deposited in international depository authorities for the purpose of own patent procedures. Currently, there are 37 depository institutions in 21 countries. In India, we have the IDA in IMTech Chandigarh, and all the culture identification, everything will be carried out there. Whenever we are doing the screening of microorganisms from some sources like wastewater or soil sample, whatever it may be, we usually following plate count method. So in the plate count method, only 1% of the microorganisms will be screened. So nowadays, the process of metagenomics is being followed for screening of novel organisms. So considering the production of enzymes, mostly these two methods will be followed. One is submerged fermentation, in which the organisms will be grown in liquid medium, and another one is solid state fermentation, otherwise called as solid substrate fermentation. So this slide shows about the production of industrial enzymes. This is the data based on association of manufacturers and formulators of enzyme products. Around 600 enzymes have been formulated and reported. Among them, 60% of the enzymes are produced from fungus alone, and 27% from bacteria, and the rest from yeast, algae, plants, and animals. So industrial enzymes, majority of the enzymes are produced from the top 5 fungal organisms, including Aspergillus, Trichoderma, Penicillium, Rhizopus fumicola, and other fungus. As we discussed, as I mentioned in the earlier slide, 27% is from bacteria and rest from other resources. So this slide shows about the global industrial enzyme market from 2013 to 24. It is expected to reach by 2024, from 2018 to 24, the demand will rise to $7.4 billion from 4.8, and with a compound annual growth rate of 5.8%. You can see how the demand is increasing. Each one indicates the application of the enzyme. For example, this blue bar, it indicates the enzymes, whatever are being used in food and beverage. Likewise, in other industries, the applications are shown. So considering the food and beverage industries, mostly alpha amylases are used. Alpha amylases are used in starch liquefaction process for the production of maltose. Now bioethanol production is of demand because of the rising price of petroleum products, and bioethanol could be produced in single step by simultaneous saccharification and fermentation using some co-cultures like Zymomonas mobilis, or some yeast and other traditional fermenting yeast also could be isolated and used. And in the textile industry, cellulases are mainly used for biopolishing of cotton fibers and alkaline proteases for degumming of silk. In laundry detergent industries, lipases are mainly used as an additive in detergent formulation to remove triglyceride soils of human sebum and fatty food stains from fabrics. Proteases are used to remove protein stains of blood, and tannases are used to remove tea stains from fabrics. Considering the oleochemical industries, mainly lipases are used. They are used for the production of fatty acids and esters. You might be knowing biodiesel, fatty acid, methyl ester, that one. And then PFOA production, polyunsaturated fatty acids, which includes EP and DHA, docosahexaenoic acid, docosahexaenoic acid. These are considered as fish oils. They are rich in fish oils. And nowadays, microorganisms are screened for production of PFOA production. There is a arachidonic acid producing fungus, namely Martirella alpina, which produces biomass of 30 to 60 gram per liter. From this, the amount of lipids could be extracted is 60%. So considering today's major problems, environmental pollution, that is mainly arising because of the transport vehicle, greenhouse gases industries, and solid and liquid waste arising from plastics, effluent from leather, detergent, textiles, food processing, and oleochemical industries. We'll be going briefly in about the plastics and leather, how the enzymes are used in the synthesis and degradation of plastics, and in what are all the steps. Being from Leather Research Institute, I have to describe what work has been carried out in our group. So we'll be focusing on this plastics area and then leather area. So plastic waste, as you all know, during this pandemic period, its usage is very much increased. Already it's known that the process in the environment for more than 400 years. India uses 8 million tons of plastic per year. Disposal by burning increases greenhouse gases. Today's plastics, they are mainly produced from non-renewable energy sources like oil, coal, and natural gas. Alternative to these conventional plastics are the biodegradable plastics or bioplastics, which could be derived from renewable resources. So bioplastics are mostly produced from plants like corn, wheat, soybeans, sweet potatoes, and sugarcane in the form of cellulose, starch, collagen, casein, and triglycerides. Starch is mainly converted into lactic acid, which is then polymerized into polylactic acid. This PLA is widely used in drug delivery and implants in the medical world. Nowadays, microorganisms are used for the production of PHS, that is polyhydroxyalkonoids. These bioplastics are comprised of biodegradable plastics and bio-based plastics. Biodegradable plastics are mainly produced from fossil materials, that they are petroleum based. This PBS, that is polybutylene succinate, polycaprolactone, and polyethyl succinate. Among these three, these two are biodegradable, depends on the organism. And BBP, bio-based plastics, could be produced from biomass and renewable resources. PHB, PLA, starch, they are easily degradable by microorganisms, but time factor is a must, because sometimes some organisms are capable of degrading the organism, even degrading the plastic even after a month's time. So various parameters are to be optimized for the degradation of these plastics. And polyethylene, nylon, and acetyl cellulose. Acetyl cellulose, based on the degree of acetylation, they are degradable or non-degradable. These are not biodegradable, polyethylene and nylon. So this polyhydroxyalkonoids or polyesters of hydroxy fatty acids synthesized as intracellular carbon and energy storage compound. They are produced in bacteria when growth is limited by nitrogen, phosphorus, or oxygen with excess amount of carbon source. The polyhydroxyalkonoids have material properties of polypropylene or polyethylene. They are natural, renewable, and biodegradable. In microorganisms, the yield of polyhydroxyalkonoids are higher, around 40 to 80%, and they exist molecular weight of 10 power 5 to 10 power 6 daltons. So bacterial synthesis of polyhydroxyalkonoids, how it occurs? The microorganisms will be utilizing or some lipids, whatever it may. Batch level in flask, and then in the fermenter they will be grown. Most of the times, not most of the time, always the polyhydroxyalkonoids will be intracellular. So the cells will be lysed and the polyplastics will be purified. After that, they will be molded and converted into products like packaging material or something like that. They are easily degradable by composting into water and carbon dioxide. So it's like a recycling process. So the metabolic pathway, how this PHB is synthesized within the organism. Generally, we'll be adding some carbohydrate like glucose or sucrose in the medium, that is in the growth medium. So those sugars will be converted into acetyl-CoA. That is, the sugars will be converted to pyruvic acid in glycolysis, and that pyruvic acid will be converted into acetyl-CoA. In the absence of oxygen, that is when there is stress in the for the microorganism, that is depletion of oxygen or nitrogen, excess phosphorus, excess carbon source. Under those cases, this pathway is getting activated. That is, the enzyme ketothiolase will convert 2 molecules of acetyl-CoA into acetoacetyl-CoA, which is then reduced to 3-hydroxybutyl-CoA, which will be polymerized to form PHB, polyhydroxybutyrate. So there is a report on production of polyhydroxybutyrate from Vibrio species, and you can see the granules of PHB, how it looks like. So this is the standard protocol. The cells were fixed and viewed under transmission electron microscope. So natural plastics degrade by all these processes. That's burning and incineration, photodegradation, vermicomposting. So enzymatic hydrolysis of polymers, how it occurs. Microorganisms, they will be secreting some extracellular enzymes. They may be lipases or esterases or cutaneous-like enzymes or depolymerases. So those enzymes attach to the surface of the polymer plastic material. They will digest the ester bond. And so by that method, some water-soluble intermediates will be formed and they will be dissolved in the medium. Later, they will be assimilated in the cells. And finally, they will be converted into carbon dioxide, water, or methane. So this slide shows about the streptomyces, which is capable of degrading polyhydroxybutyrate, polylactic acid, and polyethylene succinate. So always, whenever screening is hello. So the plastics will be emulsified some surfactant like glycer, and they will be mixed along with the medium and plated on the agar plates. So those organisms which will be showing the halogen will be selected and further studies will be carried out. This slide shows about the degradation of polybutylene succinate co-adipate. This is copolymer. This one is also copolymer, and this is polybutylene adipate. This study was carried out with commercial enzymes from Sigma. This is Pseudomonas fluorescens choline esterase (PFE), Rhizopus delimar lipase, Candida cylindrica lipase. So the polymer films were prepared by hot pressing, and then for this enzymatic degradation of Pseudomonas fluorescens esterase and Rhizopus delimar lipase, the reaction was carried out at 37 degrees centigrade. For this one, it was at 30 degrees centigrade. Meaning for these two different temperatures is all the enzymes will be having optimum activity at particular temperature and pH. So depending on their optimal activity, the reactions to be carried out. So after running the reaction, the remaining polymer films were separated and they were arranged and shown. The picture was taken. This is after 15 days of incubation. Therefore, research should be focused on isolation of microorganisms which could degrade broad range of plastics. Because like enzymes are substrate specific, even polymer degradation, sometimes they are enzyme specific, but we have to isolate an organism in such a way that the enzyme or the organism could able to degrade a broad range of plastics. Accordingly, it's to be developed. And once you, after isolating the organism, the parameters to be optimized. Under what condition, what are the things we can add for making the organism or the enzyme to make 100% degradation. So that comes under parameter optimization. And then development of a process or technology for complete hydrolysis of plastics using enzymes. So but once you do this, then the proof of concept will be available. And with that, you can scale up the process. So likewise, the process or technologies are easily developed. And in CSA or CLRA, in our laboratory, we have isolated a Cryptococcus species, which produces a novel cutaneous-like enzyme. Actually, it is we can call it as lipase, esterase, and cutaneous-like enzyme. because we are calling it as cutinase-like enzyme because its amino acid sequence matches with the cutinases of fusarium species. That's why it's also referred to as cutinase-like enzyme. This organism is unique and because of its multifaceted applications. This enzyme also shows it could be used as an additive in detergent formulation for removal of lipids, and it could be used in transesterification reaction also. It exhibits the characteristic feature of esterase, lipolytic, and depolymeric activity. Isolation of such enzyme is very rare, difficult to isolate, characterize, and to develop. We are having around 3 patents on this organism. And we received a technology award for this from Ministry of Chemicals and Fertilizers recently. Last year we received award for this. And for hydrolysis of plastics, initially whatever work we are carrying out, it's to be done in the form of emulsion. That is small aliphatic polymers like polyhydroxybutyrate or polycaprolactone, PCl. They have to be emulsified with the clysor, and then based on the decrease in turbidity of the solutions, we can find out whether how far it is degradable, something like that. So this slide shows about the degradation of different plastics like polycaprolactone, PBS, and PLA. All these plastics were purchased from Japan. The enzymes were commercially available enzymes from Amino, Fluca were used, and these are the sources: Aspergillus niger, Rhizopus oryzae. You can see the yeast, which was isolated, was capable of degrading all the plastics, polylactic acid, PBS, and polycaprolactone. While the commercial enzymes that are available in the market, they were not able to degrade these other plastics except Rhizopus oryzae, which could be able to degrade polycaprolactone. Whenever we are developing a process, the cost of the product or the cost of the process should be very cheap. So this organism initially was grown in liquid medium with trivulin as an inducer and yeast extract as organic nitrogen source. In order to make the process cost effective, we tried different agro-industrial residues for the growth and production of the yeast on agro-industrial residues. We tried different substrates. Among them, cottonseed oil cake was found to be the suitable substrate. And you can see the same image here. This one A shows the substrate matrix at 0 hours and followed by uniform growth of cells at 24 hours. And you can see the multiplication of yeast cells by budding. You can see very well in the slide. The black arrow indicates the budding cell, and the white arrow, it shows a large matured yeast cell with star. These are the star. This is due to the detachment of the cells from the matured cell. All these results were published. And we have also carried out the degradation of various films like PBSA and PBS, that is this is copolymer, polybutylene succinate coadipate, and this is polybutylene succinate. And the reaction mixer composition is shown here. The PBSA films became fragile at 8 hours. You can see fragile, that is it started broken into pieces, and it was completely degraded within 16 hours. And considering the PBS film, it turned papery only after 24 hours, and complete degradation was observed after 72 hours. We have also tried the degradation of polyurethane, which was synthesized in the lab. The polyurethane A is the control, that is without treatment, and B shows the surface of polyurethane at 24 hours with small folds. You can see small folds observed throughout the matrix of polyurethane because of the degradation by the enzyme lipase, and the fold size that is significantly increased from 48 to 72 hours. And at the end of 96 hours, there was complete degradation of polyurethane. This slide shows about the time course of polyurethane degradation by Cryptococcus species lipase. You can see around 50 milligrams of polyurethane was taken. At the end of 96 hours, it was less than 3 milligrams. And the degradation products that were analyzed were adipic acid and diethylene glycol. They were also confirmed and quantified by HPLC. This is with a bacterial strain, Rosciatilis depolymerans, which could be able to degrade aliphatic aromatic polymers. The work was carried out with PBS-10 film. PBS-10, this is polybutylene succinate, 4-terephthalate, isophthalate, and polactate. The enzymes were purified using the standard protocol. You all might be knowing the crude supernatant will be concentrated by ammonium sulfate precipitation, or it could be concentrated by ultrafiltration, depending on the molecular weight cutoff. And then there will be concentrated enzyme could be some subjected to some chromatographic techniques for purification of the proteins. So likewise, it was done, and this organism was capable of producing 2 depolymerases. It was having esterase activity, so we have named it as esterase 1 and 2. That is, one was having high molecular weight, 31 kilodalton, and another one was esterase with low molecular weight, 27 kilodalton. As I mentioned earlier, the PBS-10 film, because this is aliphatic aromatic polymer. So the plastic was made as an emulsion and degradation was carried out. You can see the reaction time A, it was done at 2 hours, B after 4 hours, C after 6 hours, and D after 24 hours. Bottle's description, all the left ones are controlled, and center one is carried out with high molecular weight esterase, and last one is with low molecular weight esterase. These work were carried out with the purified enzymes, and the enzymes added were very less, that is 2 units only added. And the degradation studies were also carried out in the form of film. You can see the disappearance of film after 72 hours with high molecular weight esterase, and some remnants we could see with low molecular weight esterase. Same work was also carried out with our yeast Cryptococcus species as a growth associated process, because whenever we are doing enzymatic process, some downstream processing steps we have to carry it out. We have to go for centrifugation and then concentration of enzyme, something like that. Some steps to be carried out. To avoid that, the degradation of these some aliphatic aromatic polymers were carried out as a growth associated process. The polymers tried were PBS-10 and PBAT. This is polybutylene adipate 4-terephthalate. This work was carried out under ambient temperature 30 degrees centigrade. Complete degradation of PBS-10 and PBAT films were observed within 96 and 216 hours respectively. SEM images confirm the formation of cracks and holes on the surface of the polymeric films during degradation. You can see how it looks like. Some round or ellipsoidal shaped cracks appeared, and the cracks evenly spread at 48 hours, and formation of big holes at 72 hours, and complete degradation was observed at 96 hours. This is with PBAT, polybutylene adipate 4-terephthalate. And after 216 hours, only complete degradation was observed. Sorry. Considering leather industry, it occupies a place of prominence in Indian economy for employment, growth, and exports. More than 60% of finished leather and leather goods are exported from India or from Tamil Nadu. Products exported are leather footwear, footwear components, leather garments, leather goods, finished leather. Considering the leather, so we are getting leather from where? From what is the source? So leathers we are getting from the raw material which are thrown away from the meat processing industry, that is hides and skins. Hides refers to outer covering of huge animals like cow, buffalo. Skins refers to outer covering of small animals like goat and sheep. So leathers are processed in 3 steps. First step is pre-tanning operation, that is we are also called as beam house process. In this process, the unwanted components like hair, flesh, fat, and soluble proteins are removed. And second step is tanning process. Here, the stabilization of the skin is carried out, and the fibers structure will be stabilized with tanning materials. Third step is post-tanning process. The tanned skins will be further processed for properties like fullness, color, softness, and finish to produce useful products. So this is the flow chart showing the beam house operations. From the slaughterhouse, the hides and skins will be brought to the tannery. They will be soaked in plain water. After that, unhairing is to be done. That is, unhairing, it refers to removal of hair. Conventionally, that is the chemical method will be followed using lime and sulfide. After this, it will be subjected to deliming to remove the lime with ammonium chloride and ammonium sulfate. After that, baiting process will be carried out to remove the soluble proteins. Then they will be subjected to pickling. After that, degreasing will be carried out with acidic. If it is like degreasing, usually in the conventional method, solvents will be used. Solvents and detergents will be used. After degreasing, they will be subjected to chrome tanning, and finally finishing will be carried out. Instead of these chemical methods, instead of using lime, sodium sulfide and other things, using enzymes, we can make the conventional method into eco-friendly method. Conventional method preservation, common salts are used. Alternative technologies, neem product and antimicrobial compounds to be used. By this method, we can preserve the hides and skins for more than 10 days. Soaking, usually water treated with some antiseptics like sodium azide or some other more products are there. And enzymatic soaking aids like proteases and carbohydrates are used. Dehairing is carried out in the tanneries using lime and sodium sulfate. Instead of this, proteolytic enzymes could be used. For degreasing, solvents and detergents will be used. This could be eliminated using lipolytic enzymes. Dehairing is mainly done to remove hair and epidermal layer. During this process, the flesh will be loosened and adipose tissue will be swollen. Commonly used proteases are from mold, Aspergillus flavus parasiticus, Aspergillus oryzae, and Coryndia bolus. From bacteria, Bacillus subtilis, amyloidofaciens, and lactobacillus. These are the known organisms. Why enzymatic dehairing? What are all the advantages? We can completely eliminate the usage of lime and sodium sulfide, and recovery of hair of good quality, creation of conducive atmosphere for the workers. Because whenever we go for the chemical method, there will be liberation of hydrogen sulfide and some eye irritation. So many problems will be created, and they are not good for the tanners who are working in the tannery. And the leather, whatever we are obtaining from the enzymatic unhairing, will be having better strength and greater surface area, and we can eliminate the baiting process, and time factor could be reduced from 18 hours, that is 6 to 12 hours. This is how the unhairing is carried out in a tannery by using lime and sodium sulfide. They will be applied as a paste on the flesh side of the hides and skin, and they will be piled one over the other, and next day with a blunt knife, the hairs will be removed. This slide shows about the application of enzyme by paint method. Here also the same. Instead of lime and sulfide, the enzyme solution will be painted on the flesh side of the hides and skins, and they will one more hide or skin will be kept on this, and they will be kept for overnight, or depending on the time factor, the process will be carried out. This is application of enzyme by spraying technique. Okay, this is the product profile of Proteus. In CLRA, we have developed a Proteus from Bacillus thymellus, and its activity is 20 plus or minus 2 units per ml. And this was partially purified by ammonium sulfate precipitation, and the activity was 750 units per gram powder. These are the application characteristics. We have shown about the optimum pH and temperature application characteristics. And this enzyme has been scaled up in our pilot plant facility from 75, 700 liter. Before that, it will be carried out in 3 liter, 7 liter, then 75, 700 liter, with working volumes of 50 and 450 liter respectively. Media components, whatever we use were fish meal and inorganic salt, simple medium. So, material cost of the medium is an important criteria, because that will add cost of the process. So better to use cheaply available resources like cornstarch liquor or fish meal or casein, something, so that we can easily reduce the cost of the media. And we have also tried on the solid substrate. This is wheat bran. Before fermentation, we inoculated the bacteria on wheat bran, and without downstream processing, the final fermented bran was dried and powdered in a homogenizer. This is the pulverized protease powder. With this powder also, deherring process could be carried out. This is deherring of goat skin. Components used were lime sulfide water enzyme. This is enzymatic method. Method of application, it could be either paste method or dip method. You can see in the conventional method how much lime is being added, 10%, 2%. In the case of enzyme assisted, see, whenever we are developing some process, we should have some standard control. Here we have used the enzyme from SPIC. The enzyme name is Bioda. So we have carried out enzyme assisted process because SPIC Bioda enzyme, it doesn't work as enzyme only process. It required a small amount of addition of lime and sulfide. In our case, in the enzyme only process, we added little amount of sodium carbonate alone, and this was the percentage of enzyme applied. And the time within 6 hours, we could be able to remove the hair from the goat skins. Similarly, the conditions are shown for the cow hides. This is enzyme assisted process, enzyme only process. Only the amount of enzyme varies with skins and hides. In the case of skins, it was 2%. In the case of hides, it was 3%. For any enzyme, for any application, the enzyme concentration or the offer level and optimum pH, temperature, all the parameters should be optimized. You can see the recovery of hair, how it looks like in the conventional method and enzymatic method. This is conventional method. A to C, it shows the hair that were obtained from the goat skin. This is conventional method. The hair was completely damaged. You cannot see the tip. And in the case of enzyme assisted method, also same thing happened. In the enzymatic method, you can see the complete recovery of hair with hair root. And this one D and E shows the cow hide. In the conventional method, we could not be able to recover the hair because it was pulped. In the enzyme only, in the enzyme assisted process, that is D, the hair was again damaged and it was recovered fully in the enzyme only process. Some studies were also carried out to see the grain surface of the hides, how it looks like. You can see remnants of short hairs are there in the hides even after unhairing in the conventional method and enzyme assisted method. Whereas in the enzyme only process, you can see the complete elimination of hair. This one shows about the opening up of collagen fiber bundle. Here it was moderate and complete opening up of fiber bundle was seen in the enzyme only process. Here there is no opening up of fiber bundle at all. And this slide shows about the dehairing with the bacillus protease. Goat skin, after 4 hours, some small hairs were sticking on the skins, and after 6 hours, complete elimination of hair. This was done with cow hide. Considering the degreasing in the chemical conventional method, solvents and detergents are used. Enzymatic degreasing, lipases could be used. These lipases could be produced from plants, animals from pancreas and microbial sources. Advantages of doing degreasing or enzymatic degreasing or elimination of solvents, reduction in surfactant, and possible recovery of valuable byproducts. Presence of why degreasing is essential. Presence of excess fat in woolly sheep skin results in various defects, like fatty spores, and even dyeing and finishing, waxy patches in alum tanned leathers, because sometimes tanning will be carried out with aluminium based tanning. So in such cases, this aluminium reacts with the grease and patches will be formed. And pink stain on wet blows. This pink stain is due to the production of pigment by Penicillium chrysogenum fungus, which will utilize the grease and some pink stains will be formed. And we have developed a lipase from Aspergillus niger, and the organism was isolated from yogurt by enrichment culture method, which was maintained on Cydia Sebactox agar plants at 4 degrees centigrade. The production of lipase was standardized in submerged fermentation as well as solid substrate fermentation. In SMF medium, the essential components were meat extract and olive oil. In order to develop a cost-effective medium, these were replaced with confit liquor and sesame oil. By using these chemicals, the material cost could be able to reduce the material cost by 60%. And this slide shows about the differences between SMF and SMF and SSF. You can see the productivity, activity, how much it is being increased in SSF. This slide shows about the scale up of lipase production from 10 grams to 100 grams to 1 kg. This is carried out in tri-substrate fermentation. See, whenever we are doing solid substrate fermentation, one substrate may not be sufficient for the growth of microorganisms. So, by adding some more substrates, say like sugarcane bagasse in powder form, or rice bran, or wheat rava, just small amount of addition of some protein-rich sources may enhance the activity by 2, 3, or 5 times like that. So all these things to be optimized. And upscaling of lipase in SSF, these are the steps to be followed in upscaling. This one shows about the autoclaved substrate in polythene bags, 1 kg each. Substrate transferred in trays after inoculation. Arrangement of loaded trays in Koji room. Trace after fermentation, you can see the black spores of the fungus of the tea, and closed views of the completely fermented substrate bed in tray. This is the scanning electron micrograph of Aspergillus niger grown on SSF. You can see tiny filamentous growth at 24 hours, followed by dense mycelial growth at 48 hours. And sorry, this is also at 24 hours. Only the difference is taken at hair magnification. And 48 hours correlation was initiated, and single sporangium was focused at 1200 hours, 72 hours. These are taken at 72 hours. And the lipase developed was used for the degreasing of sheepskin. You can see the sheepskin, how it looks like. This is before degreasing and after degreasing. So this is with reference to pre-tanning operations. During unhairing, the hair will be generated as a waste, and during degreasing, fleshings will be generated as a waste. Huge amount of solid waste will be generated during leather processing. Nearly 850 kilogram of solid waste is generated from 1000 kg of raw hides. Only 150 kilogram of the raw material is converted into leather. The solid waste generated mainly includes fleshings, 50 to 60%, foam shavings, skin trimmings, and hair. These are the raw skins. Defleshing is being carried out, and this is the fleshing waste. So these fleshings, they are very rich in proteins and fat. It contains about 70 to 80% of water. Therefore, it is essential to separate the fat and protein through suitable methods. CSIR CLRA developed an enzymatic method for extraction of lipids, and the process goes like this. The fleshings, whatever obtained from the defleshing, were de-lined to remove the lime, and after that, the de-limed fleshings was hydrolyzed by an enzymatic method. For that, we used SSF protease. This is the reaction itself. After enzymatic hydrolysis, the proteins and lipids will be in the medium, and by adding some solvent, we could separate the lipids. And this is, and that hexane also we can distillate and reuse it. And these are the separated lipids from the fleshings. And these lipids could be used in the trans-esterification reaction. So in this process, 2 enzymes have been used. One is protease, was used to prepare the protein hydrolysate and to separate the lipids. And in the second step, that is, we use lipase for converting the lipids into fatty acid methyl esters. The presence of long chain fatty acids, you all might be knowing C16 to C18 fatty acids are mainly involved in the preparation of biodiesel. They are involved in the formation of fatty acid methyl esters. So these were very rich in this lipids fleshings. Extraction of lipids, we followed different methods: protease assisted extraction, thermal hydrolysis, soft fluid extraction, and Blaine Dyer method. And maximum yield we could able to obtain in the protease assisted extraction. These are the reaction conditions. All these results were published. And after obtaining the lipids, they were characterized. You can see the fatty acid composition of the lipids. Palmitic acid stearic acid, oleic acid. So all this should not be wasted. So they were converted into fatty acid methyl esters. And if I go on presenting all the data, it will take time. So the optimum conditions for transesterification are shown here. And one more thing, because lipases are substrate specific, we cannot get theoretical yields of fatty acid methyl ester. So we can do stepwise addition of methanol, or we can use combination of lipases. In our case, we used porcine pancreas lipase, which was suitable for transesterification of lipids, and the optimum conditions were this one. But the transesterification yield was 60.33. And after that, we added Cryptococcus species lipase, and we could be able to increase the yield. And by thermal hydrolysis also, the lipids could be separated. In this slide, we have used we have shown the application of the protein hydrolysate. The protein hydrolysate, here we used thermal hydrolysis. The protein hydrolysate obtained from the enzymatic method was lyophilized, or even it could be spray dried and converted into protein-rich powder. It could be used as a supplement for the microbial growth media. Here we have called it, we are calling it as beef extract, which was used as a media component in the microbiological work. These results were published in RSA Advance, and this was used in the media component as a media component for the growth of E. coli, Pseudomonas aeruginosa, and Staphylococcus aureus. And considering the hair waste, we use the hair waste for the preparation of compost. Hair present in the hide and skin is removed, as we discussed earlier. Using lime and sulfide, the unhairing is carried out. Around 40 kilotons of hair waste is generated in India per annum. Utilization of the hair is essential because it is rich in keratin, so it will be rich in nitrogen. A bioprocess has been developed to convert the hair waste into compost within a time period of 10 to 12 days. The process provides viable technological solution to the disposal of solid waste on one hand, and provides scope for financial returns on the other. This is with reference to the Indian fertilizer demand from 2019 to 24. You can see how growth rate, how it is increasing 11.9%, and the market value will be increased by 51% from 2018 to 2024. And now the demand is farmers and public, their focus is towards organic fertilizers. So the demand will be very high in future. Organic fertilizers usage, what are all the things happening? They can be derived from plant and animal sources. They provide micronutrients for the plants and microorganisms, which maintain soil fertility. Usage of synthetic fertilizers are inorganic and lead to leaching and runoff, and they decrease the soil fertility on organic matter in the soil. So this is how we are preparing the compost at 500 kilogram level. The hair waste that are obtained from the tannery will be taken, and we isolated a keratinolytic bacterium called the Brevibacterium luteolum. Initially, the parameters were optimized for complete degradation of the hair in fast level, and then the process was scaled up to 5 liter, 50 liter, or 500 liter level in fermenter. And the keratin hydrolysate, without separating the biomass or supernatant, the whole fermentation liquor was mixed in 1:1 ratio. The following mixture comprising of oil, leaf, sawdust, and wheat bran, and they were mixed in a ribbon mixer, transferred to composting pit, and the product was packaged, and we have given the product for testing for National Agro Foundation, because all the work cannot be done in one place, and we may not be having expertise in all the areas. And the product was tested in paddy crop. It was done by National Agro Foundation. That was increase in yield of paddy crop by 20%. And this was done in chili crop. It was also around 20%. This is the overall results we have shown in this one. Apart from this, we have developed a process for decolorization of synthetic dyes. The organism was a fungal strain, Pyroneurotypa scoparia. You can see the pellets, how usually fungal spores will produce small balls. So this growth of the fungus shown in the plastic liquid medium in plastic. This was the biomass separated, and the lacase. You can see the decolorization, how it's getting done. And as growth associated process also, the process was carried out, that is the decolorization reaction. And facilities at CSAR, CLRA pilot plant. These are the facilities available in our CSAR, in our CLRA. And we are providing services to industries, tannery associations, and startup companies for demonstration of technologies. And these are our collaborators. Thank you.
Thank you, Dr. Kamini, for your lecture today. The session is open for discussion. Students, you can ask any questions, clarifications. Any questions?
I think they all will be very hungry.
Yeah. You can also use the chat box to ask questions if you want. Guess there is no questions.
No questions,
Yeah. Thank you once again, Dr. Kamini, for giving us an overview of different enzymes obtained from different sources, microbial sources, and which has wide applications like plastic degradations and applied in different steps in the process of leather processing. I hope this gives students the idea to develop their own projects based on your lecture.
Thank you very much.
So much.
Thank you.
There is one question, I think.
Yeah.
Are both protease production and protease activity proportional to each other?
What is the question?
Are both protease production and protease activity proportional to each other?
See, protease production, it will be produced in the medium. So for checking the protease activity, we have to separate the supernatant and then do the enzyme activity.
Maybe the quantity of protease produced
protease
and the activity of the protease produced. Whether there is a correlation between both.
Protease production is growth associated.
Growth associated. Okay.
Yeah.
Any other questions? Prithvi, you want to ask something? Okay, no more questions, I think. Okay, thank you so much, Dr. Kamini.
Yeah, okay, yeah. Thank you
For joining us today. Thank you. Thank you.
Thank you, sir.
Thank you.